Welcome, we're so happy you tuned in tonight. Before we get started, I'd love to know, where are you watching from? Drop it in the comments. Now, let's get comfortable and drift into deep, peaceful sleep together. Mushrooms are the fruiting bodies of certain fungi, and understanding what that means can help us appreciate these quiet organisms in a new way. When we see a mushroom growing on a forest floor or emerging from the side of a rotting log, we're looking at just one small part of a much larger living thing. The mushroom itself, the cap, the stem, the visible structure, is really just the reproductive organ of the fungus, appearing above ground or above the surface of wood when conditions are right for spreading spores. The rest of the organism, the main body of the fungus, is usually hidden from view, living beneath the soil or inside the wood where it quietly grows and feeds. Fungi belong to their own kingdom of life, separate from both plants and animals. For a long time, people grouped fungi with plants because mushrooms seem to grow from the ground like flowers or vegetables. But fungi are fundamentally different from plants in several important ways. Plants use photosynthesis to create their own food from sunlight, carbon dioxide, and water. They're producers, making energy from light. Fungi, on the other hand, cannot photosynthesize. They have no chlorophyll, no green pigment to capture sunlight. Instead, fungi are more like animals in how they obtain nutrition. They consume organic matter, breaking down food sources in their environment and absorbing the nutrients they need. This makes them heterotrophs, organisms that rely on other organisms or organic material for sustenance. But fungi accomplish this feeding in their own unique way, different from how animals eat and digest. They release enzymes into their surroundings, breaking down complex molecules outside their bodies, then absorbing the simpler compounds that result. This external digestion is one of the defining characteristics of fungi. The mushroom we see is often just the visible part of a much larger organism hidden in soil or wood. Think of it like an iceberg, where the small portion above, water, represents the mushroom, and the massive structure below represents the main body of the fungus. This hidden body is what does most of the work, absorbing nutrients, growing, surviving through seasons, and maintaining the life of the organism. The mushroom appears only when the fungus is ready to reproduce, emerging to release spores and then often withering away fairly quickly. Some mushrooms last just a few days, while others might persist for a week or two, but many are temporary structures compared to the longer-lived organism beneath. Many fungi grow as thin threads called hyphae, and these microscopic filaments are the basic building blocks of fungal structure. If you could shrink down and observe a single hypha under high magnification, you'd see a tubular cell, often just a few micrometers in diameter, with a wall made of chitin, the same material that forms insect exoskeletons and crab shells. Hyphae grow at their tips, extending forward into new territory, branching, and exploring their environment. They're surprisingly strong for their size, able to penetrate dense materials and push through soil particles. As a hypha grows, it can branch, creating new tips that spread in different directions. Some hyphae have cross walls dividing them into cellular compartments, while others form long tubes with multiple nuclei and no dividing walls. This growth pattern allows fungi to explore their surroundings efficiently, reaching toward food sources and avoiding areas that don't provide nutrition. A network of hyphae is called mycelium, and this collective structure is where the real life of the fungus happens. Mycelium can be thought of as the fungus itself, the living organism that persists year after year. When hyphae branch and interconnect, they form a dense, web-like network that can extend through soil, leaf litter, rotting wood, or any organic material the fungus feeds upon. Individual hyphae are microscopic, but together millions of these threads create a structure that can be surprisingly large. Some mycelia spread across just a few cubic inches of soil, while others extend across many square feet or even acres. The mycelium is constantly growing, dying back in places where resources are exhausted and extending into new areas where food is available. It's a dynamic system, responding to its environment with a kind of simple intelligence, growing toward nutrients, avoiding toxic substances, and adjusting to changes in moisture and temperature. Mycelium can spread through soil like fine lace or soft webbing, creating an intricate underground network. If you were to carefully sift through a handful of healthy forest soil, you might notice fine white threads running through the dark earth. These are hyphae, parts of various fungal networks threading through the spaces between soil particles. The mycelium doesn't grow in a random tangle. It tends to form organized patterns, radiating outward from a central point or spreading along favourable paths where moisture and nutrients are abundant. In some cases, mycelium forms fairy rings, circular patterns where mushrooms emerge in a ring-like formation, the result of mycelium spreading outward evenly in all directions from a central starting point. As the mycelium at the centre exhausts local food sources, the growing edge continues expanding outward, and mushrooms appear at this active frontier. These rings can expand slowly over many years, some growing just a few inches per year, while the mycelium behind the fruiting edge quietly works on decomposition. Mushrooms appear when a fungus is ready to reproduce, triggered by environmental conditions that signal the right time for spore release. Temperature, moisture, and sometimes the changing length of days can all influence when a fungus decides to produce mushrooms. Many mushrooms emerge after rain, when the soil is saturated and the air is humid, conditions that help keep the delicate fruiting body from drying out and that create favourable circumstances for spore dispersal. Some fungi fruit in spring, others in summer, and many wait for autumn when temperatures cool and rainfall increases. The timing is often species-specific, with certain mushrooms reliably appearing at the same time each year if conditions allow. When the mycelium senses that conditions are favourable, it begins to redirect resources and energy toward producing a fruiting body. Hyphae aggregate and organise themselves into the structure that will become the mushroom, forming the stem, the cap, and the spore-producing tissues in an impressively coordinated process. The main purpose of many mushrooms is to release spores, those tiny reproductive cells that will disperse and potentially start new fungal colonies elsewhere. Everything about a mushroom structure is designed to maximise spore production and dispersal. The cap protects the spore-producing surfaces beneath it from rain that might wash spores away prematurely. The stem lifts the cap up into air currents that can carry spores away from the parent mushroom. The gills, pores, or other structures on the cap's underside create enormous surface area where millions or billions of spores can be produced. A single mushroom can produce staggering numbers of spores. Some estimates suggest that a large mushroom might release trillions of spores over its lifetime. Most of these spores will never successfully establish a new fungus. They'll land in unsuitable locations, dry out, get eaten by tiny organisms, or simply fail to find the conditions they need to germinate. But by producing such enormous numbers, fungi ensure that at least some spores will survive and continue the species. Spores are tiny reproductive cells that can travel through air or water, serving as the fungus's way of reaching new locations. These microscopic structures are incredibly small, typically just a few micrometers across, small enough to remain suspended in air, currents for long periods. Spores are often protected by tough cell walls that help them survive harsh conditions. Some can endure freezing temperatures, intense heat, desiccation, and UV radiation, remaining dormant but viable until they land in a place where germination is possible. The shape, colour, and surface texture of spores vary tremendously between species, and mycologists often use these characteristics to help identify fungi. Some spores are smooth and round, others are elongated or ornamented with spines and ridges. Some are colourless, while others are pigmented in shades of brown, black, pink, or even purple. When millions of spores are released together, they can create a visible spore print, a deposit of coloured powder on any surface beneath the mushroom, useful for identification and also a beautiful reminder of the fungus's reproductive effort. A calm way to imagine a mushroom is as a forest's quiet flower, blooming briefly for spores. Like flowers, mushrooms can be beautiful, displaying diverse colours, shapes, and textures. Like flowers, they're ephemeral, appearing for a short time and then fading. And like flowers, their purpose is fundamentally about reproduction, about creating and dispersing the next generation. But unlike flowers, which rely on bright colours and sweet scents to attract pollinators, mushrooms often work more passively, relying on wind, rain splash, or gravity to move their spores. Some mushrooms do attract animals, producing odours that draw insects or releasing spores that animals will carry away on their bodies or in their digestive tracts. But many mushrooms simply stand quietly, releasing spores into the air and trusting the wind to carry them. There's something peaceful about this strategy. No urgency, no competition for pollinators, just a patient release of reproductive cells into the environment, day after day, until the mushroom withers and collapses. Now that mushrooms feel familiar, we can drift beneath the surface to meet the real everyday worker, the hidden mycelium. While mushrooms capture our attention with their visible presence, the mycelium is where the continuous work of the fungus occurs. It's the mycelium that absorbs nutrients, that grows and maintains the organism through seasons and years, that responds to environmental changes and that ultimately produces the mushrooms we see. Understanding mycelium helps us appreciate that fungi are not just the mushrooms that appear occasionally, but are persistent organisms that inhabit and shape their environments constantly. Mycelium is the main body of many fungi, the living network that persists even when no mushrooms are visible. In some ways, it's easier to understand fungi if we think of them as being like trees, where the mycelium is the trunk, branches and roots, the permanent structure, and the mushrooms are like flowers or fruit, seasonal additions that appear when conditions are right. The mycelium must gather enough energy and resources to maintain itself and to produce mushrooms when the time comes. This means that the mycelium is always working, always absorbing nutrients from its surroundings, always growing into new territory or maintaining its established network. Even in winter, where no mushrooms are visible and growth may slow or stop, the mycelium is often still alive beneath the soil or inside wood, dormant but ready to resume activity when temperatures warm and moisture returns. It can grow through soil, leaf litter, rotting logs, and even inside living roots, demonstrating remarkable versatility in where and how it lives. Soil is one of the most common habitats for mycelium, where fungal networks thread through the spaces between mineral particles and organic matter, absorbing nutrients from decomposing materials and forming relationships with plant roots. Leaf litter, the layer of fallen leaves that accumulates on forest floors, is another rich habitat full of carbon and nutrients, locked up in dead plant tissue. Mycelium can penetrate these leaves, releasing enzymes that break down cellulose and lignin, gradually reducing whole leaves to crumbly fragments. Rotting logs are particularly important habitats for many fungi. A fallen tree trunk is a massive reservoir of nutrients, and various fungal species will colonize it in succession over the years or decades it takes for the log to fully decompose. Some fungi specialize in attacking freshly fallen wood, while others prefer wood that's already partially decayed. Inside living roots, certain fungi form intimate partnerships, their hyphae actually entering root cells or forming dense sheaths around root tips, engaging in exchanges of nutrients that benefit both the fungus and the plant. Mycelium absorbs nutrients from its surroundings rather than making food from sunlight, and this fundamental difference from plants shapes everything about how fungi live. Without the ability to photosynthesize, fungi must find organic matter to consume. This makes them dependent on other organisms, either on the dead remains of plants and animals, or on living partnerships with organisms that can photosynthesize. The absorption process is efficient, but requires that the fungus be in direct contact with its food source. Hyphae grow toward nutrients, and once they reach an area rich in food, they proliferate, creating dense networks in nutrient-rich zones. The entire surface of the hyphal network can absorb nutrients, making the mycelium extremely effective at extracting resources from its environment. As nutrients are absorbed, they're transported through the hyphae to other parts of the mycelium, including to developing mushrooms when they're being produced. This transport happens through the movement of cytoplasm, the fluid contents of the fungal cells, which can flow through the hyphae in organized currents. Fungi release enzymes that break down complex materials into smaller pieces, performing their digestion outside their bodies before absorption. This is one of the most remarkable aspects of fungal biology. The enzymes that fungi produce are incredibly varied and powerful, capable of breaking chemical bonds that many other organisms cannot attack. For example, cellulose enzymes break down cellulose, the structural carbohydrate that makes up much of plant cell walls. Ligninase enzymes attack lignin, the tough, complex polymer that gives wood its strength and resistance to decay. Proteases break down proteins into amino acids. Lipases split fats into simpler components. By secreting these enzymes into dead wood, leaf litter, or soil, fungi essentially pre-digest their food in their environment, then absorb the simpler molecules that result. This external digestion means that fungi can tackle food sources that would be impossible for many other organisms to use. Dead wood, for instance, is nutritious, but locked away inside tough lignocellulose structures. Without fungi and bacteria to break it down, dead wood would persist almost indefinitely in many environments. Those smaller pieces can then be absorbed through the hyphae, completing the feeding process. Once enzymes have broken down large molecules into smaller ones, sugars from cellulose and starch, amino acids from proteins, fatty acids from lipids, these smaller molecules can pass through the cell walls and membranes of the hyphae. Fungi use active transport and other cellular mechanisms to move these nutrients into their cells, where they can be used for energy and growth. The efficiency of this process is remarkable. Fungi can extract nutrients from materials that seem completely used up, gleaning nutrition from sources that other organisms have already exploited. This scavenging ability makes fungi essential to ecosystem nutrient cycling, ensuring that elements like carbon, nitrogen, and phosphorus don't remain locked up in dead organic matter but instead return to forms that can be used by plants and other organisms. Mycelium can be very large, spreading over wide areas when conditions are right. In fact, some of the largest organisms on Earth are fungi, their mycelia extending across enormous areas. One famous example is a honey fungus, Armillaria orstoii, in Oregon's Blue Mountains, where genetic testing revealed that a single individual mycelial network extends across more than 2,000 acres, making it one of the largest and oldest organisms known, estimated to be thousands of years old. Most mycelia are much smaller than this record holder, but even modest fungal colonies can be surprisingly extensive. A mycelium the size of a dinner plate might contain miles of hyphae when all the threads are added together. This extensive growth allows fungi to access resources across a broad area, connecting different patches of nutrients and transferring resources within their network. The mycelium can respond to local conditions, growing more densely where food is abundant and extending exploratory hyphae into new areas where nutrients might be found. Because it's hidden, a forest can contain far more fungus than we can see. When you walk through a forest, the visible mushrooms represent just a tiny fraction of the fungal biomass present. Most of the fungal life is invisible, woven through the soil and wood all around. Studies that have analysed forest soil have found astounding amounts of fungal tissue. In some temperate forests, the mass of fungal hyphae in the soil can exceed the mass of all the tree roots. In a single teaspoon of healthy soil, there might be miles of fungal hyphae threading through the particles and pore spaces. This hidden abundance underscores how important fungi are to forest ecosystems. They're not occasional visitors or minor players. They're fundamental, constant presences that shape soil chemistry, influence plant health, and drive the recycling of nutrients throughout the ecosystem. Mycelium often grows slowly and steadily, responding to moisture and food sources with patient persistence. While some fungi can grow quite rapidly under ideal conditions, you might notice a new patch of mycelium appearing on a log or spreading across compost within days. Most growth in natural environments is more measured. Hyphae extend at their tips, adding length bit by bit, hour by hour. The rate of growth depends on temperature, moisture, and the availability of food. Warm, moist conditions with abundant nutrients allow faster growth. Cold, dry or nutrient-poor conditions slow growth considerably. Unlike animals that move to find food, mycelium stays put and grows toward resources. If you could watch a time-lapse of mycelium exploring a new substrate, you'd see hyphae extending and branching like slow-motion tendrils, searching for the best path forward, avoiding obstacles, and concentrating growth in the most rewarding areas. It can pause growth during dry times and continue when rain returns, showing a remarkable resilience. Fungi are well adapted to the variable conditions of many environments. When water is scarce, mycelium can become dormant, essentially waiting for better conditions. The hyphae might dry out and cease active growth, but they don't necessarily die. When rain returns and moisture penetrates the soil or wood where the mycelium lives, the fungus can rehydrate and resume growing, sometimes remarkably quickly. This ability to pause and resume activity allows fungi to persist through droughts, cold snaps, and other challenging periods. It also means that mushroom appearances can be unpredictable. After a long dry spell, a sudden heavy rain can trigger a flush of mushrooms as mycelia that have been waiting for moisture suddenly spring into reproductive mode. This pattern gives forests and grasslands their rhythms of fungal fruiting, periods of abundance alternating with times when few or no mushrooms are visible. Walking through a forest, you're often stepping over a quiet, living network. Every footfall lands on ground that's lightly threaded with mycelium. The top few inches of soil, the decomposing leaf litter, the rotting logs, all are inhabited by fungi going about their slow work. This realization can change how you perceive a forest. It's not just the trees, ferns, birds and insects. It's also this vast, mostly invisible community of fungi interconnected with the plant roots, breaking down the dead, feeding on the fallen, and creating the conditions that allow the visible forest to thrive. There's something humbling and comforting about this idea, that beneath the apparent stillness of the forest floor, there's constant activity at a microscopic scale, a biological engine quietly running, sustaining the ecosystem's cycles. With that underground calm in mind, let's explore fungi's gentle job as recyclers, turning old wood and leaves into fresh nutrients. Decomposition is one of the most essential ecological functions, and fungi are among the most important decomposers on the planet. Without decomposition, dead organic matter would accumulate endlessly, nutrients would be locked away in unusable forms, and ecosystems would grind to a halt. Fungi make it possible for death to transform into new life, completing the cycles that keep forests, grasslands, and other ecosystems healthy and productive. Many fungi are decomposers, meaning they break down dead organic material and return its components to the ecosystem. These decomposer fungi are sometimes called saprotrophs or saprophytes, terms that describe their lifestyle of feeding on dead matter. They're specialists at decay, equipped with enzymes that can dismantle the complex molecules in dead plants and animals. This is different from parasitic fungi, which feed on living hosts, and from symbiotic fungi, which form mutually beneficial partnerships. Decomposer fungi are recyclers, and their work is neither harmful nor helpful to individual organisms. It's simply essential to the ecosystem as a whole. They process leaves, wood, animal remains, and other organic debris, converting it into simpler substances that can re-enter the nutrient cycles that support new growth. Fallen leaves, dead trees, and old plant matter are all food sources for decomposer fungi. In autumn, temperate forests shed enormous quantities of leaves, creating a fresh blanket of organic matter on the forest floor. As these leaves lie on the ground, they become colonized by fungi and bacteria. Fungal spores land on the leaves, germinate, and send hyphae into the leaf tissue. Over weeks and months, the leaves gradually lose their structure, becoming softer, darker, and more fragmented. This is fungal decomposition in action. Dead trees, whether standing snags or fallen logs, provide a different kind of resource. Huge amounts of woody material that can take years or decades to fully decompose. Different fungi colonize wood at different stages. Some fungi attack recently dead trees, while others specialize in wood that's already partially rotted. The succession of fungal species on a decomposing log is a well-studied phenomenon, with certain mushrooms appearing only on fresh logs and others only on very rotten, crumbly wood. Fungi help return nutrients like carbon and nitrogen to the soil, making these elements available for uptake by plants and other organisms. Carbon, which plants fix from the atmosphere through photosynthesis, becomes incorporated into plant tissues, cellulose, lignin, proteins, and other compounds. When the plant dies, this carbon would remain locked in the dead tissue if not for decomposers. Fungi break down these carbon-containing molecules, respiring some of the carbon back into the atmosphere as carbon dioxide and incorporating some into their own bodies and into the soil as decaying organic matter. Nitrogen, a key nutrient that plants need but that's often in short supply, also gets locked up in dead plant and animal tissues. Fungal decomposition helps release nitrogen from proteins and other organic molecules, converting it into forms like ammonium that plants can absorb through their roots. This nitrogen cycling is crucial to maintaining soil fertility. Without decomposers, forests would pile up with undecomposed material, creating an ecosystem crisis. Imagine a forest where dead leaves never rotted, where fallen trees never decayed. Over time, the forest floor would become choked with layers and layers of intact dead plant material. Nutrients would become increasingly scarce in the soil because they'd all be tied up in this accumulating debris. New plant growth would slow and eventually cease for lack of available nutrients. Of course, this doesn't happen because decomposers, primarily fungi and bacteria, constantly work to break down dead material at roughly the same rate that it's produced. In healthy ecosystems, there's a balance between the input of dead organic matter and its decomposition. This balance maintains soil fertility and keeps ecosystems productive over the long term. Fungi can break down tough plant materials such as cellulose. the structural carbohydrate that makes up a large part of plant cell walls. Cellulose is made of glucose molecules linked together in long chains, and these chains are packed tightly together in plant tissues, making cellulose very stable and resistant to degradation. Most animals cannot digest cellulose directly. Even animals that eat plants, like cows and deer, rely on symbiotic bacteria and other microorganisms in their digestive systems to break down cellulose for them. Fungi, however, produce cellulose enzymes that can cleave the bonds between glucose molecules in cellulose, breaking the long chains into shorter segments and eventually into individual glucose molecules. This glucose can then be absorbed and used by the fungus for energy and growth. This ability to digest cellulose means that fungi can thrive on plant materials that many other organisms cannot use, giving them access to a huge reservoir of potential food in the form of dead leaves, grass, wood, and other plant debris. Some fungi can also break down lignin, a strong component of wood that's even tougher than cellulose. Lignin is a complex, irregular polymer that gives wood much of its strength and rigidity. It's also what makes wood resistant to decay. Lignin is so stable that it doesn't break down easily even under harsh conditions. Only certain fungi, notably white rot fungi and brown rot fungi, have evolved the enzymatic machinery to degrade lignin. White rot fungi can completely break down both lignin and cellulose, leaving behind pale, soft, spongy wood. Brown rot fungi primarily attack cellulose, leaving behind brown, crumbly wood that's rich in modified lignin. The ability to degrade lignin is ecologically crucial because it allows the vast amounts of carbon locked up in woody tissue to be recycled. Without lignin-degrading fungi, dead wood would persist almost indefinitely, and forests would be choked with ancient unrotted logs. This ability helps logs gradually soften and return to the earth, transforming wood into soil. A fallen tree trunk begins as solid, hard wood, but over years and decades fungal activity transforms it. The wood becomes softer, darker, and more porous. It begins to crumble when touched. Eventually it loses all structural integrity, collapsing into a pile of humus that enriches the soil. This transformation is not a violent or sudden process. It's slow, gentle, and thorough. As the log decays, it also becomes a habitat. Insects bore into the softening wood, birds excavate nests, small mammals find shelter in cavities, and plants send roots into the moist, nutrient-rich wood. A decomposing log supports a succession of life even as it breaks down, and by the time it's fully decayed, it has nurtured countless other organisms and contributed its stored nutrients back to the soil. Decomposition is usually slow, matching the quiet pace of forests. Unlike the rapid decomposition that occurs in hot, moist tropical environments or in compost heaps, forest decomposition in temperate and boreal regions is often measured in years. A leaf might take 1 to 3 years to fully decompose depending on its toughness and the local climate. A small branch might take 5 or 10 years. A large log might persist for 20, 50, or even more than 100 years before it's fully incorporated into the soil. This slow pace is partly due to temperature. Fungal and bacterial activity slows in cold conditions, and partly due to moisture availability. It's also a function of the toughness of the material being decomposed. Needles from pine trees, rich in resins and waxy coatings, decompose more slowly than leaves from deciduous trees. Hardwood logs, dense and full of lignin, take longer to decay than softer woods. Fungi often work alongside bacteria and small insects in recycling ecosystems, forming decomposer communities. Decomposition is rarely the work of a single organism. Instead, it's a collaborative effort involving many species. Bacteria are particularly important in breaking down soft, nitrogen-rich materials like animal carcasses and green plant tissue. Fungi excel at breaking down tougher, carbon-rich materials like wood and dry leaves. Together, bacteria and fungi complement each other's abilities, creating more complete and efficient decomposition than either group could achieve alone. Insects, mites, springtails, and other tiny invertebrates also play important roles. They physically fragment dead material, chewing leaves and wood into smaller pieces that have more surface area for microbial colonization. Some insects feed on fungi themselves, dispersing fungal spores and helping fungi colonize new substrates. This web of interactions means that decomposition is a community process, not just a fungal one, though fungi are often the key players. In a gentle way, Fungi help forests tidy up without rushing. There's no urgency in decomposition, no deadline to meet. The forest produces dead material, leaves in autumn, branches after storms, trees at the end of their lifespans, and the decomposer community processes this material at its own pace. The pace is dictated by temperature, moisture, and the nature of the material itself. If conditions are cold or dry, decomposition slows. If it's warm and wet, it speeds up. But there's a balance, a rhythm that persists year after year. The forest doesn't accumulate endless debris, nor does everything disappear instantly. Instead, there's a layered structure to the forest floor, fresh leaves on top, partially decomposed material below, and fully humified organic matter mixed with mineral soil at the bottom. This structure reflects the progression of decomposition, with materials at different stages coexisting peacefully, each contributing to the forest's health in its own time. Now we can look at a calmer kind of partnership, when fungi don't just recycle, but cooperate with living plants and trees. Not all fungi are decomposers living on the dead. Many have evolved relationships with living plants, forming partnerships that are among the most important ecological relationships on Earth. These partnerships, called mycorrhizae, connect the lives of fungi and plants in ways that benefit both partners and shape entire ecosystems. Many fungi form partnerships with plant roots called mycorrhizae, a term that literally means fungus root. In these relationships, fungal hyphae associate closely with plant roots, sometimes forming a sheath around the outside of the root, sometimes penetrating between or even into root cells. These are not parasitic relationships where one partner benefits at the expense of the other. Instead, they're mutualistic, meaning both the fungus and the plant gain something valuable. The prevalence of mycorrhizae is remarkable. It's estimated that more than 90% of plant species form mycorrhizal associations. This means that when you look at a forest, a meadow, or even a garden, almost every plant you see is connected underground to a fungal partner. This partnership has deep evolutionary roots, likely dating back hundreds of millions of years to when plants first colonized land. Some scientists suggest that mycorrhizal fungi may have been essential to that colonization, helping early plants extract nutrients from barren mineral soils. In mycorrhizae, fungal threads connect with or enter a plant's roots, creating an intimate physical connection. There are several types of mycorrhizae, but the two most common are ectomycorrhizae and arbuscular mycorrhizae. In ectomycorrhizae, which are common in temperate forest trees like pines, oaks, and birches, the fungus forms a dense sheath of hyphae around the outside of fine root tips. Hyphae penetrate between root cells but don't usually enter the cells themselves. This creates a large interface between fungus and root where nutrients can be exchanged. In arbuscular mycorrhizae, which are common in grasses, crops, and many herbaceous plants, the fungus sends hyphae that penetrate the root and actually enter some of the root cells, forming highly branched structures called arbuscules inside the cells. These arbuscules are where the exchange of nutrients takes place, with the plant and fungus trading resources across the cell membrane. The fungus can help the plant absorb water and minerals from the soil, extending the plant's reach far beyond its own roots. Fungal hyphae are much finer than even the finest plant root hairs, allowing them to penetrate tiny soil pores that roots cannot access. The hyphae also extend much farther from the plant than roots typically do. A single plant's mycorrhizal network might extend through hundreds of cubic inches, or even cubic feet of soil, exploring far more area than the plant's roots could cover on their own. This extended reach is especially important for absorbing nutrients that don't move easily through soil, particularly phosphorus. Phosphorus is essential for plants. It's a key component of DNA, ATP, and other critical molecules, but it's often scarce in soil and doesn't diffuse readily through water. Mycorrhizal fungi can seek out phosphorus across a broad area and transport it back to the plant, making it available even when soil phosphorus levels are very low. The fungi also help with water absorption, gathering moisture from a larger volume of soil and helping the plant stay hydrated during dry periods. The plant can share sugars it made through photosynthesis with the fungus, providing the food energy that the fungus needs. This is the plant's side of the bargain. Because fungi cannot photosynthesize, they need a source of carbon-containing compounds to fuel their growth and metabolism. The plant, which is using sunlight to convert carbon dioxide and water into sugars, has an abundant supply of these compounds. In a mycorrhizal partnership, the plant transports some of its photosynthetically produced sugars, primarily glucose and other simple sugars, down to its roots and then transfers them to the fungus. The fungus uses these sugars for energy and to build more hyphae, which in turn gather more nutrients for the plant. In some forest ecosystems, it's estimated that plants may transfer 10% or more of the carbon they fix through photosynthesis to their mycorrhizal partners. That's a substantial investment, but one that pays off in improved nutrition and survival. This trade benefits both partners when conditions are right, creating a relationship of mutual support. The plant gets enhanced access to nutrients and water, which can improve its growth, health, and ability to survive stress like drought or nutrient deficiency. The fungus gets a steady supply of sugars and other carbon compounds, which it cannot make for itself. Both partners gain something they need, something they could not obtain as easily on their own. This kind of mutualism is common in nature, but mycorrhizae are particularly important because they're so widespread and because they have such significant effects on plant performance and ecosystem function. In some cases, plants can grow without mycorrhizal partners, but they often grow more slowly and are more susceptible to stress. In other cases, certain plants are essentially dependent on mycorrhizae and will not thrive, or may not even survive, without their fungal partners. Mycorrhizal fungi can extend the effective reach of a plant's root system, sometimes dramatically so. Research has shown that the hyphal networks of mycorrhizal fungi can increase the absorptive surface area of a plant by hundreds or even thousands of times. If you were to measure all the surface area of a tree's roots and root hairs, and then add in all the surface area of the mycorrhizal hyphae connected to those roots, the total absorptive area would be vastly greater than the roots alone. This expanded network allows the plant to tap into nutrient and water reserves that would otherwise be inaccessible. It's like having an enormously extended root system without having to invest the energy and resources to grow all those roots. The plant gets the benefits of a much larger root system, but shares the cost with the fungus, which grows the hyphae and absorbs nutrients that benefit both partners. These partnerships are common in forests and grasslands, shaping the structure and function of these ecosystems. In forests, most trees form ectomycorrhizal or arbuscular mycorrhizal associations. The fungi involved are often species that produce the mushrooms we see on the forest floor: boletes, chanterelles, russulas, amanitas, and many others. These mushrooms are the fruiting bodies of the mycorrhizal fungi that live in partnership with tree roots. When you see a mushroom growing near a tree, there's a good chance it's connected, underground to that tree's roots, part of a mycorrhizal network. In grasslands, arbuscular mycorrhizae dominate, supporting the grasses and herbaceous plants that make up these ecosystems. The fungi involved in arbuscular mycorrhizae rarely produce large mushrooms. Their spores are usually distributed in soil or by animals, and the fungi themselves are mostly invisible, known primarily through microscopic examination of roots and soil. Different plant species often associate with specific types of mycorrhizal fungi, showing preferences or compatibility patterns. Some trees, like pines, primarily form partnerships with specific groups of ectomycorrhizal fungi. Other trees, like maples, form associations with different fungal groups. Some plants are generalists, forming partnerships with many different fungal species, while others are more selective. These preferences can influence forest composition and diversity. If a particular tree species needs a particular mycorrhizal fungus to thrive, the presence or absence of that fungus can determine whether the tree can successfully establish in a given area. In some cases, foresters have found that when planting trees in new areas, inoculating the soil with appropriate mycorrhizal fungi can greatly improve tree survival and growth. This practical application of mycorrhizal science demonstrates how important these partnerships are. This relationship can support healthier plant growth and better nutrient uptake, with measurable benefits. Studies comparing plants with and without mycorrhizal fungi consistently show that mycorrhizal plants tend to grow larger, produce more biomass, and have better access to nutrients, especially phosphorus and nitrogen. Mycorrhizal plants often show improved drought tolerance, possibly because the extensive hyphal network helps them access water during dry periods. Some research also suggests that mycorrhizal fungi can help protect plants from certain soil-borne diseases, either by outcompeting pathogenic organisms or by enhancing the plant's own defense mechanisms. These benefits can translate into better crop yields in agriculture, healthier trees in forests, and more resilient plant communities in natural ecosystems. The relationship is not always beneficial. Under some conditions, the cost to the plant of supporting the fungus may outweigh the benefits, but in most situations the partnership is mutually advantageous and contributes to plant success. It's a quiet example of cooperation, exchange, support, and steady balance. Mycorrhizae remind us that competition is not the only force shaping ecosystems. Cooperation and mutualism are equally important. The plant and fungus have different strengths and different needs, and by partnering, they can both succeed more fully than either could alone. There's no aggression in this relationship, no domination. It's a trade, a collaboration, an interweaving of lives at the smallest scales that has profound effects on entire landscapes. This partnership has persisted for hundreds of millions of years, shaping the evolution of both plants and fungi and making possible the green, productive ecosystems we see today. With that partnership glowing softly in the background, let's return to the mushroom itself, its parts, shapes, and gentle variety. Mushrooms are incredibly diverse in form, and understanding their structures helps us appreciate both their beauty and their function. Each mushroom's shape reflects its strategy for producing and dispersing spores, and the variety of solutions fungi have evolved is a testament to the creative power of natural selection. Many mushrooms have a cap and a stem, but not all do. The classic mushroom shape, a rounded cap atop a central stem, is familiar and common, but it's just one of many forms. Some mushrooms have no stem at all, growing directly attached to wood or other substrates. Some are shaped like shelves or brackets projecting horizontally from tree trunks. Others are round like puffballs or shaped like coral, antlers, cups, or crusts. The form a mushroom takes is closely related to how it produces and releases spores and to the environment in which it grows. A mushroom growing on the forest floor might benefit from an elevated cap to catch wind currents, while a mushroom growing on the side of a vertical tree trunk might produce spores that fall away from the tree into open air. The diversity of forms is one of the delights of mushroom hunting. You never know quite what shape the next fungus might take. The underside of the cap often has gills, pores, or teeth-like structures, and these features define major groups of mushrooms. Gilled mushrooms, which include many familiar species, have thin, blade-like structures radiating from the stem out to the edge of the cap. These gills are covered with spore-producing cells, and spores form on the surfaces of the gills, eventually dropping into the space between gills and falling, away from the mushroom. Pored mushrooms, including the boletes, have sponge-like undersides covered with tiny holes. Each hole is the opening of a tube, and spores are produced inside these tubes, dropping out through the pores. Toothed fungi have downward-pointing spines or teeth, and spores form on the surfaces of these spines. Each design maximizes surface area in its own way, creating ample room for the production of countless spores. These structures increase surface area for producing spores, which is key to the mushroom's reproductive success. If a mushroom had a flat underside, the area available for spore production would be limited to a circle the size of the cap. But by adding gills, pores, or teeth, the mushroom creates complex three-dimensional surfaces that have far more total area than a flat surface would. Gills, for example, create many vertical surfaces, each one covered with spore-producing cells. The total surface area of all the gills combined can be many times the area of the cap itself. This maximization of surface area allows a single mushroom to produce enormous numbers of spores, sometimes billions from a single fruiting body. This abundance is necessary because spore survival rates are very low, and the mushroom must produce vast quantities to ensure that at least a few spores will successfully germinate and establish new mycelia. Gilled mushrooms release spores from the edges and faces of their gills, a process that's both elegant and efficient. The gills are oriented vertically, which allows spores to drop straight down into the spaces between the gills under the influence of gravity. Specialized cells called basidia, located on the gill surfaces, produce spores at their tips. When mature, the spores are forcibly ejected from the basidia, a process driven by tiny water droplets that propel the spore outward. This ejection is on a microscopic scale. The spore is shot just a fraction of a millimeter, but it's enough to clear the gill surface and enter the space between gills. Once in that space, the spore falls downward, dropping out from under the cap into the open air. If the mushroom is positioned well, air currents can then catch the spore and carry it away. This system works best when the gills are perfectly vertical, which is why you'll often see mushrooms adjusting their growth to keep the cap level, even when growing on sloped ground. Some mushrooms have pores instead of gills, like many boletes, creating a different but equally effective spore release mechanism. Boletes are easily recognised by their spongy undersides, which are actually masses of tiny vertical tubes packed closely together. Each tube is lined with spore-producing cells, and spores drop down through the tubes and out the pores at the bottom. This design is particularly common in mushrooms that form mycorrhizal relationships with trees. The advantage of pores over gills is durability. The spongy mass is less fragile than delicate. Gills and may be better suited to certain conditions. Boletes are often quite substantial mushrooms, and the thick, spongy pore layer can produce truly staggering numbers of spores over the course of the mushroom's life. The pores also allow for steady, continuous spore release, as spores drop out as soon as they mature, without needing to be forcibly ejected. Some mushrooms are shaped like shelves and grow on tree trunks, adapting to their vertical environment. These shelf fungi, also called bracket fungi or polypores, are often tough and woody rather than soft and fleshy. They project outward from the sides of trees, and their undersides, facing downward, are covered with pores or gills from which spores drop. Growing on vertical surfaces offers certain advantages. The mushroom doesn't have to compete for space on the crowded forest floor, and it's positioned right next to its food source if the fungus is decomposing the tree's wood. Many shelf fungi are perennial, growing a new layer of tubes or pores each year, building up a thick layered structure over time. Some of these mushrooms can persist for years or even decades, becoming hard and hoof-like, slowly releasing spores season after season as they break down the tree they're growing on. Puffball mushrooms release spores in little clouds when disturbed, a striking and memorable method of dispersal. Puffballs are round or pear-shaped, and as they mature, their interior tissue breaks down into a mass of powdery spores. When the outer skin dries and cracks, or when something, a raindrop, a passing animal, a curious human, taps or steps on the puffball, spores puff out in a visible cloud, like smoke or dust. This cloud can contain millions of spores, carried away on the slightest breeze. The puffball mechanism is particularly effective in dry conditions, when wind is available to disperse spores over long distances. There's something playful about puffballs. Finding a mature one and tapping it to release a cloud of spores is a small joy that many people remember from childhood walks in the woods. Some fungi produce bright colours, while others blend into forest browns and greys, and this variation serves different purposes. Brightly coloured mushrooms, reds, oranges, yellows, purples, can be warnings to potential predators that the mushroom is toxic or unpalatable. The vivid red of Amanita muscaria, for instance, is iconic and serves as a signal. Other bright mushrooms may use colour to attract animals that will help disperse spores, though this is less common than in flowering plants. Many mushrooms, however, are cryptic in coloration: browns, tans, greys, whites, blending into the leaf litter or wood on which they grow. These colours may help the mushroom avoid being eaten, or simply may not confer enough advantage for bright pigmentation to evolve. The colours themselves are produced by various pigments, and can be useful for identifying mushroom species, as certain pigments are characteristic of particular groups. A mushroom's shape often reflects how it spreads spores, tying form to function in beautiful ways. Tall mushrooms with elevated caps catch more wind. Spreading caps create shade beneath, maintaining the humidity needed for delicate gills. Puffballs enclose spores until the right moment, then release them all at once. Shelf fungi use gravity, letting spores drop away from the tree into open space. Stinkhorns produce foul-smelling slime that attracts flies, which then carry spores away on their bodies. Birds' nest fungi form tiny cup-shaped structures with eggs inside, splash cups that fling spore-containing packets when raindrops hit them. Each strategy is an evolutionary solution to the challenge of getting spores from the parent mushroom out into the environment where they might start new colonies. The mushroom's diversity is like a quiet wardrobe of nature. Many styles, one purpose. Whether gilled or pored, bright or drab, elevated or ground-hugging, each mushroom is ultimately doing the same thing, releasing spores. The diversity arises from the many different ways evolution has found to accomplish this goal under varying conditions. In a single forest, you might find dozens or even hundreds of different mushroom species, each with its own shape, colour, habitat preference, and timing. This diversity is not chaotic, but is instead a collection of finely tuned adaptations, each species occupying its own ecological niche, each contributing to the overall function of the ecosystem in its own particular way. Now we'll drift into the gentle life cycle, how spores travel, settle, and begin new hidden networks. The life of a fungus doesn't end when the mushroom withers. It begins again each time a spore successfully germinates. Understanding the fungal life cycle helps us see mushrooms not as isolated individuals, but as parts of ongoing lineages, each generation contributing to the persistence of the species across time and space. Spores are usually microscopic and very lightweight, perfectly designed for long-distance travel. A typical fungal spore might measure just a few micrometers in diameter, far too small to see with the naked eye. At this size, spores are easily lifted and carried by air. They have very little mass, so they can remain suspended in air currents for long periods, potentially travelling miles from the parent mushroom. The lightweight nature of spores is crucial to fungal dispersal. If spores were heavy, they would simply fall to the ground near the parent mushroom, leading to competition and little expansion of the species' range. But because they're so light and so numerous, spores can spread far and wide, colonizing new areas and ensuring that the fungal species can survive even if conditions become unsuitable in one location. Wind can carry spores through forests like invisible dust, distributing them broadly across the landscape. On a breezy day in autumn, the air in a forest is full of fungal spores, though we cannot see them. Studies that sample air in different environments consistently find fungal spores present, sometimes in high concentrations. These spores are part of the natural background of particles in the air, along with pollen, bacteria, and mineral dust. Wind dispersal is passive but effective. Spores simply drift wherever the wind takes them, and because they're so small, they can travel long distances before settling. Some spores may travel just a few feet, landing near the parent mushroom. Others may be carried hundreds or thousands of feet, crossing meadows, roads, or rivers to land in entirely new habitats. Some spores travel by water splashes or along damp surfaces, an alternative dispersal strategy suited to certain environments. When raindrops hit a mushroom or the ground near one, they can splash spores into the air or carry them along the surface of soil or leaves. This splash dispersal is common in mushrooms that grow close to the ground or in wet environments. Some fungi produce sticky spores that adhere to water droplets, ensuring they're carried along when water moves. In very wet habitats, like mossy forests or areas near streams, water dispersal can be as important as wind dispersal. Spores might be washed into streams and carried downstream, or they might be splashed from one leaf to another, gradually moving through the environment in tiny liquid-borne steps. Animals can also help move spores on fur, feet, or by eating mushrooms, creating yet another dispersal pathway. Some mushrooms produce odours that attract insects, slugs, or other small animals. These animals may feed on the mushroom tissue, ingesting spores in the process. The spores can pass through the animal's digestive system and be deposited elsewhere in its feces, potentially in a nutrient-rich package that helps the spore germinate. Other animals simply brush against mushrooms and pick up spores on their fur or feathers, carrying them to new locations. Larger animals like deer, squirrels, and birds can transport spores over considerable distances this way. Some fungi, especially underground truffles, rely almost entirely on animal dispersal. Truffles produce strong scents that attract animals like rodents, pigs, and deer, which dig up and eat the fruiting bodies, dispersing the spores in their droppings. When a spore lands in a suitable place, it can germinate into a new fungal thread, starting the cycle anew. Germination requires the right conditions: sufficient moisture, appropriate temperature, and a substrate that can provide food. If these conditions are met, the spore absorbs water, swells, and begins to grow. A tiny hypha emerges from the spore, extending into the surrounding environment. This first hypha is the beginning of a new mycelium. It grows, branches, and starts to explore its surroundings, seeking nutrients. If conditions remain favourable, the hypha will continue to grow, forming a network that can persist and expand for years or even centuries. This is how fungi colonize new areas, one spore at a time, germinating and establishing new mycelia wherever conditions allow. That thread can grow and branch, forming new mycelium that may one day produce its own mushrooms. The young mycelium starts small, just a few branching threads in the soil or wood. But over time, as it absorbs nutrients and grows, it can become extensive. Months or years after germination, the mycelium may be ready to reproduce. When conditions are right, the right temperature, moisture, and nutrient availability, the mycelium begins to form mushrooms, aggregating hyphae into the structures that will become stems and caps. The mushrooms mature, produce spores, and release them, completing the cycle. The mycelium that produced those mushrooms may continue to live and grow, producing more mushrooms in future seasons, or it may die back if conditions become unfavourable. Either way, the cycle continues, linking generations of fungi through the dispersal and germination of spores. Many fungi require compatible partners to combine genetic material before forming certain mushrooms, adding complexity to reproduction. Fungi have various reproductive strategies, and many species have multiple mating types. For sexual reproduction to occur, two compatible mycelia must meet and fuse. This fusion combines genetic material from both partners, creating genetic diversity in the resulting spores. The fused mycelium can then produce mushrooms that contain this mixed genetic material, and the spores from these mushrooms will carry new combinations of genes. This sexual reproduction is important for generating variation, which allows fungi to adapt to changing conditions and resist diseases. Some fungi can also reproduce asexually, producing spores that are genetically identical to the parent. This can be useful for rapidly colonizing suitable habitats, but it doesn't create the genetic diversity that sexual reproduction does. Fungal reproduction can be complex even when the forest feels simple. From the surface, a forest seems straightforward: trees, shrubs, animals, and the quiet passage of seasons. But beneath this apparent simplicity is tremendous complexity in the reproductive strategies and life cycles of fungi. Some fungi have 2 or more nuclear phases in their life cycles. Some have multiple types of spores, produced in different ways at different times. Some can switch between sexual and asexual reproduction depending on conditions. This complexity is hidden from casual observation, revealed only through careful study. But it's this complexity that allows fungi to be so successful, so adaptable, so resilient across diverse environments and changing conditions. The visible mushroom is often brief, but the mycelium can persist much longer, a reminder of where the real life of the fungus resides. A mushroom might last a few days or a few weeks, but the mycelium that produced it can live for years, decades, or longer. Some individual mycelia are thought to be thousands of years old, persisting underground, slowly expanding, fruiting occasionally when conditions are right. This longevity means that fungi are not just the mushrooms we see. They're the persistent networks that endure through time, connecting past and future through their continuous presence in the ecosystem. When you see a mushroom, you're seeing a brief expression of a much older, much larger life form, a temporary ambassador from the hidden world beneath your feet. In a comforting way, fungi remind us that most important work happens quietly in the background. The mushrooms are beautiful and fascinating, but they're just the tip of the iceberg. The real work, the decomposition, the nutrient absorption, the partnerships with plant roots, the slow spreading through soil, happens out of sight, day after day, year after year. This is true in many areas of life. The visible, dramatic moments are supported by countless unseen efforts. Fungi teach patience and humility. They show that steady, quiet persistence can accomplish vast things over time, and that you don't need to be loud or showy to be essential. With those tiny spore journeys in mind, let's explore how fungi fit into whole ecosystems, supporting soil, plants, and small creatures. Fungi are not isolated organisms. They're deeply embedded in the ecosystems where they live. Their activities influence soil structure, plant health, animal populations, and the cycling of nutrients. Understanding these ecosystem roles helps us appreciate fungi not just as interesting organisms, but as critical players in the natural world. Fungi help build healthy soil by breaking down organic matter into usable nutrients, a foundational ecosystem service. Soil is not just ground-up rock, it's a complex mixture of minerals, organic matter, water, air, and living organisms. The organic component of soil, the dark, crumbly humus, is largely the product of decomposition. As fungi and bacteria break down leaves, wood, and other plant material, they create humus, a stable form of organic matter that improves soil structure, holds nutrients, and supports plant growth. Humus gives soil its fertility, its ability to retain moisture, and its characteristic dark color. Without the decomposing action of fungi, soils would be far less productive. The nutrients locked in dead plant material would remain unavailable to living plants, and the soil would lack the organic matter that gives it structure and fertility. This process contributes to the rich smell of forest soil after rain, a scent that many people find pleasant and evocative. That earthy smell is partly due to compounds released by soil microbes, including fungi. One compound often associated with this scent is geosmin, produced by certain bacteria, but fungi contribute their own volatile compounds to the complex bouquet of forest soil. The smell is a signal that the soil is alive and active, full of organisms working to break down organic matter and cycle nutrients. It's the smell of decomposition, not as something unpleasant, but as something vital and generative. Breathing in that scent after a rain, you're experiencing the chemical signature of the ecosystem's recycling process. Fungi can help soil hold moisture by improving structure over time, particularly through the production of hyphae and sticky compounds. Fungal hyphae bind soil particles together, creating small aggregates that give soil structure. These aggregates create pore spaces in the soil, tiny gaps where air and water can move. Good soil structure means that water can infiltrate rather than running off, and that the soil can hold moisture in its pores, making it available to plant roots over time. Some fungi produce glomalin, a sticky protein that further stabilizes soil aggregates. Over time, the activity of fungi and other soil organisms creates a soil with good tilth, easy to work, able to absorb and hold water, and resistant to erosion. This improved soil structure is one of the less visible but highly important contributions fungi make to ecosystem health. Many insects and small animals feed on fungi or live near fungal growth, creating food webs centered on fungal resources. Fungal fruiting bodies, mushrooms, are food for a wide range of organisms. Slugs and snails graze on mushroom tissue. Insects lay eggs in mushrooms, and their larvae feed and develop inside the fruiting bodies. Some beetles specialize in feeding on particular types of fungi. Springtails and mites feed on fungal hyphae in the soil. Even vertebrates like squirrels, deer, and small rodents eat mushrooms, sometimes selectively seeking out certain species. This means that fungi support animal populations not just by recycling nutrients, but by serving directly as food. In some ecosystems, the seasonal appearance of mushrooms is an important food source for wildlife, providing protein and nutrients at critical times. Some animals rely on fungi as part of their diet, either occasionally or as a primary food source. Northern flying squirrels, for example, feed heavily on truffles and other underground fungi, and they play a role in dispersing the spores of these fungi through their feces. Some insects have obligate relationships with fungi. They cannot complete their life cycles without fungal food sources. Certain beetles vector fungal spores to new logs, where the fungi decompose the wood and provide food for beetle larvae. In tropical regions, leafcutter ants cultivate fungi in their nests, feeding the fungi with fresh-cut leaves and then eating the fungal tissue. These intricate relationships show how deeply fungi are woven into the fabric of ecosystems, not just as background organisms, but as central players in food webs and ecological interactions. Fungi can also influence which plants thrive in a given area through their partnerships, shaping plant community composition. If a particular plant requires a particular mycorrhizal fungus to grow well, the distribution of that fungus can determine where the plant can successfully establish. Conversely, if a plant can form partnerships with many different fungi, it may be more flexible and able to grow in a wider range of conditions. Some invasive plants succeed partly because they can form partnerships with local mycorrhizal fungi, gaining the benefits of these relationships as soon as they arrive in a new area. On the other hand, some rare or endangered plants may struggle in part because their specialized mycorrhizal partners are also rare or have declined. Understanding these fungal-plant relationships is increasingly recognized as important for conservation and restoration, as reintroducing the right fungi can sometimes help re-establish native plant communities. Forest ecosystems depend on these underground relationships for stability, with mycorrhizal networks linking trees and sharing resources. Recent research has revealed that mycorrhizal networks can connect multiple plants, even plants of different species, creating what some have called a wood wide web. Through these networks, nutrients and even chemical signals can move between plants. A large, well-established tree might share sugars with smaller shaded seedlings through fungal connections, potentially helping the next generation survive in low light. If a tree is attacked by insects, it might send chemical signals through the mycorrhizal network that prompt neighbouring trees to increase their own defences. These findings suggest that forests are more interconnected than previously thought, with fungi playing a central role in linking individual plants into a cooperative community. When conditions change, fungal communities can shift too, responding to alterations in climate, moisture, or plant composition. If a forest experiences a drought-tolerant fungi may become more common while moisture-loving species decline. If tree composition changes, for example, if a forest transitions from oaks to pines, the mycorrhizal fungi associated with those trees will shift accordingly. If temperatures rise or rainfall patterns change, fungal species that thrive in the new conditions may replace species that thrived in the old conditions. These shifts can happen gradually, over years or decades, and they can have cascading effects on the plants and animals that depend on fungal. Understanding how fungal communities respond to environmental change is an active area of research, with implications for predicting how ecosystems will respond to climate change and other disturbances. Even so, fungal roles, recycling and partnering, remain steady themes in nature, constants amid the changes. No matter what species of fungi are present, the basic functions remain the same. Dead material gets decomposed and recycled. Living plants form partnerships with fungi to improve nutrient uptake. These processes have been happening for hundreds of millions of years and will continue as long as there are plants and fungi. The specific players may change, but the play remains the same. This constancy is reassuring. It speaks to the resilience of ecosystems and to the fundamental importance of fungi in the natural world. A forest can be seen as a community above ground, supported by a quieter community below. The trees, shrubs, wildflowers, birds and mammals are the visible members of the forest community, the ones we notice and appreciate. But beneath them is another community, the soil organisms, the decomposers, the mycorrhizal networks, that makes the above-ground community possible. This below-ground community is just as diverse and just as important, even though it's mostly invisible. The two communities are connected, each depending on the other, each shaping the other. Fungi are key members of the below-ground community, and understanding their roles helps us see forests as integrated wholes rather than just collections of trees and animals. Now we'll gently touch on humans and mushrooms, not as rules or warnings, but as calm notes about curiosity and study. Humans have long been fascinated by mushrooms, and our relationship with fungi is complex, encompassing food, medicine, culture, and science. Approaching this relationship with curiosity and respect can deepen our appreciation for fungi and their place in the world. People have studied mushrooms for centuries in many cultures, recognizing both their practical uses and their intrinsic interest. In ancient times, people learned which mushrooms were safe to eat and which were poisonous, knowledge passed down through generations. Some cultures developed sophisticated mushroom cultivation techniques, growing fungi for food long before modern agriculture existed. In Europe, truffle hunting became an art, with trained animals helping people locate the prized underground. Fungi. In Asia, certain mushrooms were valued not just as food, but as medicine, used to treat various ailments. Indigenous peoples around the world have incorporated mushrooms into their diets, ceremonies, and healing practices, developing deep knowledge of local fungal diversity. The scientific study of fungi is called mycology, a field that encompasses everything from the molecular biology of fungal cells to the ecology of fungal communities. Mycologists identify and classify fungi, study their life cycles, investigate their biochemistry, and explore their roles in ecosystems. The field has grown enormously in recent decades as new molecular techniques have allowed scientists to detect and identify fungi that don't produce visible fruiting bodies or that are otherwise difficult to study. DNA sequencing has revealed that fungal diversity is far greater than previously thought, with thousands of species still unnamed and unstudied. Mycology is a vibrant field full of discoveries and unanswered questions, appealing to scientists interested in everything from ecology to genetics to biochemistry. Scientists classify fungi by genetics, structure, and life cycles, creating a taxonomy that helps organize the immense diversity of fungal life. Early classifications were based on visible characteristics: the shape of the mushroom, the color of the spores, the structure of the gills or pores. As microscopy improved, scientists began to examine spore shapes, hyphal structures, and other fine details. More recently, genetic analysis has revolutionized fungal taxonomy. By comparing DNA sequences, scientists can determine evolutionary relationships between fungi, grouping species that are closely related and separating those that only look similar but are not closely related. This genetic approach has led to many reclassifications and has clarified the fungal tree of life, showing how different groups of fungi are related and how they have evolved over time. Some mushrooms are used as food, while others are not eaten, and distinguishing between them requires knowledge and caution. Edible mushrooms have been part of human diets for millennia. Mushrooms like button mushrooms, shiitakes, oyster mushrooms, and portobellos are cultivated commercially and widely available. Wild mushrooms like chanterelles, morels, and porcini are prized by foragers and chefs for their flavors and textures. But many wild mushrooms are inedible, and some are dangerously toxic. Eating the wrong mushroom can cause illness or even death, so proper identification is critical. People who forage for wild mushrooms often spend years learning to identify species reliably, using field guides, attending workshops, and seeking guidance from experienced foragers. The rule, when in doubt, throw it out, is wise. No mushroom meal is worth the risk of poisoning. Fungi are also used in making bread rise, brewing, and fermenting foods, though those are yeasts rather than typical mushrooms. Yeasts are single-celled fungi, lacking the complex multicellular structures of mushrooms, but they're fungi nonetheless, and they're enormously important to human culture and cuisine. Yeasts ferment sugars into alcohol and carbon dioxide, processes harnessed for making beer, wine, and bread. Baker's yeast makes dough rise, creating the light texture of bread. Brewer's yeast ferments the sugars in grain or fruit, producing alcohol and the flavors of beer and wine. Other fungi are used in fermenting foods like soy sauce, miso, tempeh, and certain cheeses. The blue veins in blue cheese, for instance, are colonies of mycelium mold. These culinary fungi are so integrated into human foodways that it's hard to imagine cuisine without them. Some fungi produce compounds used in medicine and research, contributing to human health and scientific knowledge. The most famous example is penicillin, the antibiotic derived from Penicillium mold, which revolutionized medicine and saved countless lives. Other fungi produce antibiotics, immunosuppressants, cholesterol-lowering drugs, and other medically important compounds. Some mushrooms produce compounds being studied for potential anti-cancer, anti-inflammatory, or immune-modulating effects. Fungi are also important model organisms in biological research. Yeast cells, for example, are used extensively in genetic and molecular biology studies because they're simple, easy to grow, and share many fundamental cellular processes with human cells. Insights gained from studying fungi have broad applications in medicine, agriculture, and biotechnology. Fungal enzymes are studied for their ability to break down tough materials, with potential applications in industry and environmental cleanup. The same enzymes that allow fungi to decompose wood and other plant materials can be harnessed for human purposes. Cellulose and ligninase enzymes, for instance, are being explored for breaking down agricultural waste, producing biofuels, and improving paper manufacturing. Some fungi can break down pollutants like petroleum hydrocarbons, pesticides, and even plastics, leading to interest in using fungi for bioremediation, cleaning up contaminated sites naturally. Research into fungal enzymes is ongoing, with scientists seeking to understand how these enzymes work and how they might be used to solve practical problems. Mushrooms can be grown in controlled environments for study and cultivation, making fungi accessible for research and food production. Cultivating mushrooms allows scientists to study fungal biology in detail, controlling variables and observing growth and reproduction under different conditions. It also provides a sustainable source of food. Mushroom cultivation can be done on a small scale, in a home garden or even indoors, or on a large industrial scale. Some mushrooms are easier to cultivate than others. Button mushrooms and oyster mushrooms grow readily on composted materials or straw. Shiitakes grow on logs or sawdust blocks. Other mushrooms, particularly mycorrhizal species, are more difficult or impossible to cultivate because they require living plant partners. But for those that can be grown, cultivation offers a way to enjoy mushrooms without relying on wild foraging and a way to study fungi year-round. Learning about fungi often reveals how connected ecosystems truly are, opening our eyes to relationships we might not have noticed. when you start to understand that most plants have fungal partners, that dead wood is habitat and food for countless organisms, that mushrooms are just visible moments in long underground lives, the forest becomes a different place. You see connections instead of isolated organisms. You notice the cycling of nutrients, the flow of energy, the partnerships and dependencies that link life forms together. This ecological perspective is one of the great gifts of studying fungi. They're such integral parts of ecosystems that learning about them means learning about the systems themselves. Fungi invite a slow kind of curiosity. Notice, observe, and appreciate the quiet work. There's no need to rush when learning about fungi. They're patient organisms, and they reward patient observation. You might visit the same fallen log week after week, watching as different mushrooms appear and fade as the wood gradually softens and crumbles. You might photograph mushrooms, noting their shapes and colours, trying to identify them later. You might simply sit quietly in the forest and think about the hidden networks beneath your feet, the silent decomposition, the slow exchanges between roots and hyphae. This kind of engagement is meditative, calming, and enriching. It asks nothing but your attention and offers in return a deeper connection to the natural world. To finish, we'll bring the forest's recycling team back to bedtime scale. Soft transformations, slow cycles, and a calm mind. Fungi teach lessons that extend beyond ecology into how we might think about our own lives. Lessons about patience, about the value of quiet work, about cycles of growth and decay, about cooperation and connection. Decomposition is not a harsh ending. It's a quiet return of materials to the soil, a transformation rather than a termination. When a leaf falls or a tree dies, it doesn't simply vanish. It becomes something else: food for fungi, habitat for insects, nutrients for future plants. This transformation is gradual and gentle, a slow yielding to the processes of decay. There's no violence in it, just chemistry and biology working at their own pace. Thinking about decomposition in this way can be comforting. Endings are not really endings, they're transitions, returns, contributions to the continuation of life. Fungi help transform fallen trees into nutrient-rich ground for new growth, completing the cycle that allows forests to renew themselves. A tree that lived for 100 years, accumulating carbon and nutrients, eventually falls. Over the next few decades, fungi and other decomposers will dismantle that tree, releasing its stored resources back into the ecosystem. Where the tree once stood, new seedlings may grow, nourished by the soil enriched with nutrients from the fallen giant. This is succession at work, the process by which ecosystems change and renew themselves over time. Fungi are central to this process, the agents of transformation that allow old structures to become new possibilities. This cycle supports future plants without needing loud change, operating on nature's own timeline. There are no announcements, no sudden shifts. The cycle simply continues day by day, year by year. Seeds germinate, plants grow, plants die, fungi decompose, nutrients return to soil, new seeds germinate. The cycle is ancient and reliable, requiring no intervention, no management, no haste. It's a system that works because each part does its job in its own time. This patience is built into nature, and observing it can help us find patience in our own lives. Many forest nutrients are reused again and again through these processes, creating a kind of natural economy. In ecosystems, waste is rare because everything is eventually recycled. The carbon that was once in a tree might become part of a mushroom, then return to the air as CO2, then be taken up by a new tree through photosynthesis. The nitrogen that was in a fallen leaf might be absorbed by a fungus, passed to a plant through a mycorrhizal connection, incorporated into new leaves, and eventually return to the soil again when those leaves fall. This reuse is efficient and sustainable, a model of circularity that human systems often aspire to but rarely achieve. Mycelium can link different parts of the forest through shared soil spaces, creating physical connections that integrate the ecosystem. The hyphal networks spread through soil, touching roots, decomposing organic matter, exploring pore spaces. These networks can be extensive, connecting trees, shrubs, and herbaceous plants in an underground web. Through these connections, the forest becomes more than a collection of individual plants. It becomes a community, interlinked and interdependent. This connectedness is invisible from the surface, but it shapes how the forest functions, how nutrients move, how plants support each other. Mushrooms appear and fade, but the fungal network can remain, a reminder that persistence doesn't require constant visibility. We often equate presence with visibility, but fungi teach us that the most enduring aspects of an organism can be hidden. The mushroom is temporary, but the mycelium endures. We might draw parallels to our own lives. The visible accomplishments, the public moments, are supported by the quieter, ongoing work we do day after day. Resilience and persistence often happen beneath the surface, in the steady, unglamorous work that sustains us over time. Rain often encourages mushroom growth because fungi thrive in moisture, responding to environmental cues with precise timing. A dry spell might mean no mushrooms for weeks, but a good rain can trigger a flush of fruiting within days. This responsiveness shows how attuned fungi are to their environment, how they wait for the right conditions rather than forcing growth when times are unfavourable. There's wisdom in this, knowing when to act and when to wait, responding to conditions rather than ignoring them. The smell of damp forest earth can be linked to microbial life and decomposing matter, a sensory reminder of the hidden activity. That rich, earthy scent is the smell of life in transformation, of organisms breaking down and building up, of the forest's respiratory and digestive systems at work. It's a smell that many people find comforting, perhaps because it's associated with forests, with nature, with places that feel alive and whole. Breathing in that scent, we're taking in molecules released by fungi and bacteria, tangible evidence of the invisible work happening all around us. Fungi show that slow change can be healthy and natural, offering an alternative to the speed and urgency that often dominate modern life. We live in a culture that values speed, that wants results now, that can become impatient with processes that take time. Fungi remind us that many of the most important processes cannot be hurried. Decomposition takes the time it takes. Mycelium grows at its own pace. Mushrooms appear when conditions are right, not when we demand them. This natural slowness is not laziness or inefficiency. It's the appropriate pace for the work being done. Accepting this can reduce anxiety and help us align ourselves with natural rhythms rather than fighting against them. In a cozy way, fungi teach that letting go can be part of making space for new calm. Just as a fallen tree must decompose to make room and resources for new growth, we sometimes need to let go of things, old habits, outdated beliefs, finished projects, to make space for new possibilities. Holding on tightly to everything, refusing to let anything end or change, creates stagnation. Letting go, allowing natural conclusions, accepting endings as part of a cycle rather than as failures, these can bring a sense of calm and renewal. Fungi, in their patient work of breaking down and transforming, model this graceful approach to endings. Now we'll settle everything gently, like a forest floor at dusk, quiet, soft, and ready for rest. As the day fades in a forest, light dims, temperatures cool, and the forest settles into evening. The birds grow quiet, the breeze calms, the forest floor, covered in leaves and moss, becomes a soft bed where small creatures nestle and where fungi continue their invisible work. There's a peacefulness to this transition, a sense of the day releasing its grip and night arriving gently. We can invite that same gentle settling into our own experience, letting thoughts quiet, tensions ease and rest. Approach softly. Mushrooms are brief appearances of a much larger hidden life below, ephemeral expressions of persistent organisms. When you remember this, mushrooms take on new meaning. They're not isolated objects, but connected to vast networks, brief moments of visibility and long lives lived mostly underground. This perspective can shift how we see many things, recognizing that what's visible is just a small part of a larger whole, that there's always more happening beneath the surface. Mycelium works quietly, absorbing, sharing, and recycling day after day without fanfare. It doesn't seek attention or recognition. It simply does its work, responding to conditions, growing where growth is possible, forming partnerships, breaking down materials. This quiet competence is admirable. There's no drama, no crisis, just steady function. In our own lives, much of what sustains us happens just as quietly. The routines we maintain, the small kindnesses we offer, the daily work we do. This quiet work is valuable, even essential, though it may never be celebrated. Forests stay healthy partly because fungi keep nutrients moving, preventing depletion and maintaining productivity. The cycling of nutrients is fundamental to ecosystem health, and fungi are key drivers of this cycling. By breaking down dead material and releasing nutrients, by transferring nutrients between plants through mycorrhizal networks, fungi ensure that resources remain available and that ecosystems don't run down over time. This maintenance work, invisible but crucial, is what allows forests to persist for centuries and millennia. The mushroom's cap and gills are simple tools for spore release, evolved solutions to the challenge of reproduction. There's elegance in this simplicity, no unnecessary complexity, just the structures needed to do the job. The cap protects the gills maximize surface area, the spores drop and drift away. It works, and it has worked for millions of years. Sometimes, simple solutions are the best ones, refined through time until they're perfectly suited to their purpose. Spores travel lightly, trusting wind and water to carry them, surrendering to forces beyond their control. A spore has no agency, no ability to steer or choose its destination. It simply releases and drifts, trusting in numbers and chance. Most will fail, but some will find the right spot, germinate, and continue the cycle. There's a kind of faith in this process, a trust in the mechanisms of dispersal that have worked for countless generations. We too must sometimes trust processes we cannot control, releasing our grip and allowing outcomes to unfold. Mycorrhizal partnerships show that cooperation can be as important as competition, balancing the narratives we often hear about nature. While competition and survival of the fittest are real and important, cooperation and mutualism are equally prevalent and equally significant. Fungi and plants cooperate, sharing resources, supporting each other, creating outcomes that benefit both partners. This cooperation is not altruistic in the human sense. It's driven by mutual benefit, but it's cooperation nonetheless, and it shows that success in nature often comes not from outcompeting others but from finding ways to work together. The forest's calm stability comes from many small processes happening slowly, an emergent property of countless interactions. No single organism or process makes the forest stable. Instead, stability emerges from the integration of many parts. Decomposition here, growth there, partnerships everywhere, nutrients cycling, water percolating, energy flowing. Each small process contributes to the whole, and the whole is far more stable and resilient than any single part. This is how complex systems work, and it's a comforting reminder that stability doesn't require perfection or control. It arises naturally from healthy interconnection. Thinking about mushrooms can soften the mind, like watching mist settle on leaves, gently redirecting attention to quiet, natural processes. There's something inherently calming about mushrooms and fungi. They're not threatening or aggressive. They don't demand anything. They simply are, quiet, patient, doing their work in their own time. Contemplating them, imagining mycelium spreading through soil, picturing spores drifting on wind, visualizing the slow transformation of a fallen log. These thoughts can quiet mental chatter and invite a meditative state, a soft focus that allows the mind to rest. Tonight, you can let your thoughts decompose gently, breaking into smaller, quieter pieces, just as fungi break down complex molecules into simpler ones. Thoughts that are tangled or troubling can be allowed to fragment, to lose their sharp edges, to soften and dissolve. There's no need to hold on to thoughts tightly. They can drift apart, their components released, their energy dispersed. In the same way that fungi transform a fallen tree into soil, you can allow your thoughts to transform into something quieter, something more restful, something that nourishes rest rather than preventing it. And you can rest, knowing the world knows how to renew itself, softly and patiently, in cycles that have continued for eons and will continue long after tonight. The forest doesn't need your vigilance to keep functioning. The fungi will continue their work. The mycelium will grow. The cycles will turn. You are part of these cycles, connected to them through breath and food and the shared chemistry of life, but you don't have to manage them or control them. They go on, reliably, and you can trust in that continuation. You can release the day, release your concerns, and allow yourself to settle into sleep, knowing that the world is taking care of itself, that you're held by systems far older and wiser than worry, that rest is natural and necessary and safe. Take a slow inhale, and then a longer exhale, and imagine your mind like a quiet forest floor, soft, warm, and gently settling into sleep. The forest floor is forgiving, supporting whatever rests upon it without judgment. It's made of countless small pieces, leaf fragments, crumbled wood, fungal threads, all loosely connected, all part of a whole. Your mind can be like this, soft and accepting, each thought just a small fragment settling into place, no thought too heavy or too sharp, everything gradually coming to rest. The warmth of the forest floor accumulated from the day's sun and held in the organic matter is the warmth of being alive, of being held, of being part of something larger. And as you settle, as your breathing slows and deepens, as your body relaxes into the support beneath you, you can imagine yourself becoming part of that quiet forest floor, sinking into rest as naturally as a leaf falling, as gently as a spore landing, as peacefully as the forest itself settling into the stillness of night. The next story is about to begin. Please relax and enjoy. Coral reefs are underwater ecosystems built largely by corals over long periods of time. When you picture a coral reef, you might imagine bright colours, busy fish, and intricate structures rising from the ocean floor. But at its heart, a reef is something quieter than that initial impression suggests. It's a construction project that has been underway for thousands of years, built by creatures so small you could hold dozens of them in your palm. These structures don't appear overnight. They accumulate slowly, steadily, one tiny addition at a time, like a wall being built one grain of mortar at a time, one small stone placed carefully next to another. The reef grows in silence, in darkness, and in light, beneath waves that roll overhead without hurrying the process along. Corals themselves are animals, not plants, even though they can look plant-like when you see them swaying gently in the current or branching upward toward the light. This surprises many people at first. The stationary nature of corals, their rootedness to one spot, their sometimes bushy or tree-like appearance, all suggest something botanical. But corals are members of the animal kingdom, related more closely to jellyfish and sea anemones than to any seaweed or kelp. They don't produce their food through photosynthesis alone, though as we'll see, they have partnerships that involve photosynthesis. They capture food from the water, they respond to their environment. They are, in the quietest sense, hunters and builders at the same time. Many reef-building corals live in colonies made of many tiny individuals. Picture an apartment building where each unit is occupied by a nearly identical resident, all working together without meetings or conversations, simply performing their individual tasks in a way that benefits the whole structure. Each coral colony can contain hundreds, thousands, even millions of individual coral animals, and yet the colony functions as a kind of unified whole. There's no central brain directing operations, no manager deciding where the next addition should be built. The organisation emerges from simple rules followed by each tiny individual, repeated across the colony, repeated across time. Each tiny coral animal is called a polyp. This word might sound technical, but it simply refers to the individual unit of coral life. A polyp is small, often just a few millimetres across, though some species have larger polyps. If you could shrink down and observe a single polyp up close, you would see a simple body structure, a tubular form, a mouth surrounded by tentacles, and a base that connects to the skeleton below. The polyp is soft-bodied, vulnerable on its own, but it doesn't live on its own. It lives in community, surrounded by other polyps that are often genetically identical clones of one another, all budded off from parent polyps in a slow process of colony expansion. Polyps often have tentacles that help them catch tiny food from the water. These tentacles can extend outward, waving gently in the current like small fingers reaching into the passing water. The tentacles are equipped with specialized stinging cells, similar to those found in jellyfish, though typically much milder. When a piece of plankton drifts close enough, the tentacles can capture it, draw it toward the polyp's mouth, and begin the process of digestion. This feeding often happens at night, when many types of plankton are more active in the water column, and when the polyp can extend its tentacles more fully without the risk of being disturbed by fish or other daytime activity. Reef-building corals make hard skeletons, usually from calcium carbonate. This is the same material found in chalk, in limestone, in seashells. The coral polyp secretes this material at its base, building a cup-like structure called a corallite, within which the polyp sits. The skeleton is external, unlike the internal skeletons of vertebrates. The polyp lives on top of its skeleton, continually adding to it from below, lifting itself slightly higher over time. The calcium carbonate is drawn from the seawater itself, where calcium ions and carbonate ions are dissolved. The polyp facilitates a chemical reaction that brings these ions together into a solid mineral form, depositing it in precise layers that form the architecture of the reef. Over time, these skeletons accumulate and form the reef structure. When a polyp dies, its skeleton remains. New polyps can settle on top of old skeletons, adding their own layers. Colonies grow upward and outward, branching and mounding according to their species' growth pattern. The reef becomes a layered record of life and growth, with the living tissue forming only a thin veneer on the surface, and beneath it, countless layers of skeleton produced by generations of polyps that lived years, decades, centuries ago. The reef is, in this sense, mostly made of the past. The present is only the newest, thinnest layer, but it's the layer that continues the building process. Coral reefs are often found in warm, shallow, clear ocean waters. Temperature matters because corals, like many marine creatures, are sensitive to the temperature of their environment. Most reef-building corals thrive in water that stays between roughly 23 and 29 degrees Celsius, warm enough to support their metabolism and the metabolism of their algal partners, but not so hot that stress begins to set in. Shallow water matters because light is essential for many reef-building corals, due to their relationship with photosynthetic algae. Light penetrates best in the upper layers of the ocean, where the water is only a few meters to a few dozen meters deep. Clear water matters for the same reason. Sediment and particles in the water scatter and absorb light, reducing the amount that reaches the coral and its algae. In murky water, the partnership that powers reef growth becomes less efficient. Reefs can support a huge variety of life in a relatively small area. Walk through a tropical forest and you will find immense biodiversity, but the biodiversity of a coral reef, measured per square meter, can rival or even exceed that of the forest. Fish of every shape and size weave through the coral branches. Invertebrates cling to surfaces, burrow into crevices, filter water, scavenge scraps. Algae coat the rocks, seagrasses may grow nearby, sponges rise in colourful tubes and mounds. The three-dimensional structure of the reef creates niches, small and large, each one a potential home or feeding ground. The complexity of the habitat allows for complexity in the community, a principle familiar from ecosystems on land but especially concentrated in the reef environment. A calm way to picture a reef is as a slow-growing city built one small room at a time. Imagine a city where construction never stops, but also never rushes. Each building is added brick by brick with no machinery, no cranes, just the patient work of tiny builders who do the same task every day. The city grows outward and upward, its layout determined not by a master plan but by the accumulated decisions of countless small actors. Some neighbourhoods are dense and intricate, others are more open and sturdy. The city provides housing, food sources, shelter from danger. It becomes a hub for activity, attracting residents from far and wide. And yet, if you sit quietly and watch, the growth is almost imperceptible. You would need to return year after year to notice the change. Now that we've met the reef itself, we can drift closer to the coral polyps, those tiny builders working quietly together. Understanding the polyp is essential to understanding the reef, because the polyp is both the living organism and the architect of the structure. It is easy to think of the reef as a static thing, a geological feature, but in truth it is the cumulative result of biological activity. Every curve, every branch, every pore in the reef's surface was shaped by a living polyp or a colony of polyps responding to their environment. A coral polyp is a small animal related to jellyfish and sea anemones. All three belong to a group called cnidarians, a name derived from the Greek word for nettle, referring to the stinging cells these animals possess. Cnidarians have a simple body plan compared to many other animals. They lack a brain, though they have a nerve net that allows them to sense and respond to stimuli. They lack complex organs, relying instead on a single body cavity that serves as a gut. Despite this simplicity, they have been successful for hundreds of millions of years, adapting to environments from the open ocean to tidal pools to the deep sea. Polyps live inside a cup-like part of the skeleton they build. This cup, the corallite, is tailored to the polyp's body. The polyp sits within it, protected on the sides and bottom, exposed at the top where its mouth and tentacles can interact with the water. The skeleton provides support and defense, a permanent home that the polyp never leaves. As the polyp grows, it adds to the skeleton's base, raising itself slightly. The corallite grows with the polyp, maintaining the protective relationship throughout the polyp's life. Many corals grow by budding, where a polyp makes a new polyp beside it. This asexual reproduction is one of the primary ways that coral colonies expand. A polyp will begin to grow a new polyp from its side, a process that starts as a small bulge in the tissue and gradually develops into a complete functional polyp with its own mouth, tentacles, and skeleton. The new polyp remains connected to the original, sharing tissue and sometimes even sharing resources through internal connections. Over time, this budding creates large coral colonies, where hundreds or thousands of polyps are all interconnected, all genetically identical clones of one another. Over time, this budding creates large coral colonies that can span meters across. A single colony might start from a single larva that settled on the reef and grew into a founding polyp. That polyp budded off new polyps, which budded off more, and the colony grew in a pattern determined by the species' genetics. Some corals form branching shapes, with polyps adding new branches that reach upward and outward. Others form mounding or dome shapes, growing more slowly but creating massive sturdy structures. Still others form plate-like or encrusting shapes, spreading across the substrate like a living pavement. Each shape has advantages in different environments, and the diversity of forms contributes to the reef's structural complexity. Polyps can extend their tentacles, especially when feeding. During the day, many coral polyps keep their tentacles retracted, pulling them in close to the body to reduce the risk of being nipped by passing fish, or to reduce water resistance in strong currents. But at night, when the water is calmer and plankton is more abundant, the tentacles extend outward, creating a feeding net. The extended tentacles increase the polyp's feeding area, allowing it to capture more food. In some coral species, the transformation from day to night is striking. A smooth, closed surface during the day becomes a fuzzy, intricate field of waving tentacles after dark. Coral polyps can catch plankton and tiny particles drifting in the water. Plankton includes a wide variety of organisms and organic material. Microscopic algae, tiny shrimp-like creatures called copepods, larvae of various marine animals, bits of detritus. When these items drift close to a coral polyp's tentacles, the stinging cells, called nematocysts, can fire, injecting a small barb and sometimes a mild toxin that immobilizes the prey. The tentacles then bend inward, bringing the captured food to the polyp's mouth. Digestion occurs within the polyp's body cavity, and nutrients are absorbed. Waste is expelled back through the mouth, the only opening in the polyp's simple body plan. Many corals also receive energy from a partnership with tiny algae. This partnership is one of the defining features of reef-building corals, and it explains much about where reefs form, how they grow, and why they are so productive in otherwise nutrient-poor tropical waters. The algae are single-celled organisms, photosynthetic, capable of using sunlight to produce sugars and other organic compounds. These algae live inside the coral tissue, within the cells of the polyp, in a relationship that benefits both partners. The coral provides a safe environment and access to sunlight. The algae provide the coral with a portion of the energy they produce through photosynthesis. Coral growth is usually slow, measured in centimeters per year for many species. Some branching corals can grow relatively quickly, adding several centimeters per year under good conditions. Massive mounding corals grow more slowly, sometimes only a few millimeters per year. This slow pace means that large coral structures represent many years, often many decades, sometimes even centuries of growth. A massive coral head the size of a car might be hundreds of years old, a living record of countless polyp generations, countless meals captured, countless layers of skeleton deposited in the quiet darkness beneath the polyp's tissue. Despite slow growth, reefs can become massive over long time spans. The Great Barrier Reef, for example, stretches over 2,000 kilometers along the coast of Australia, making it the largest living structure on Earth. It didn't form overnight. It formed over thousands of years, beginning when sea levels rose after the last ice age and flooded the shallow continental shelf. Corals settled on the newly submerged rock grew, reproduced, died, and were built upon by new corals. The reef today is the product of this patient, ongoing process, a structure so large it can be seen from space, built by animals so small you could fit dozens on a coin. Reef strength comes from patience and repetition, not speed. There is something calming in this fact. something that runs counter to the pace of much of modern life. The reef doesn't rush, it doesn't cut corners, it doesn't try to grow faster than conditions allow. It simply continues day after day, year after year, adding small increments that accumulate into something vast and enduring. The strength of the structure comes not from any single polyp or any single year of growth, but from the sheer accumulation of effort over time, the way a mountain is worn down grain by grain, or the way a canyon is carved drop by drop. With those tiny builders in mind, let's explore the reef's quiet, secret helper, the algae that live with corals in a gentle partnership. This relationship, called symbiosis, is central to understanding how coral reefs can thrive in tropical waters that are often very low in nutrients. In the open ocean, nutrients like nitrogen and phosphorus are often scarce in the sunlit surface waters, having been consumed by plankton or settled into the deep. And yet, coral reefs are among the most productive ecosystems on Earth. The key to this productivity is the partnership between coral and algae, a relationship that allows the coral to capture energy from sunlight, in addition to capturing food from the water. Many reef-building corals host microscopic algae inside their tissues. If you could slice through a coral polyp and examine it under a microscope, you would see the algae nestled within the cells of the coral's inner layer, called the gastrodermis. The algae are tiny, single-celled organisms, golden brown in colour due to their photosynthetic pigments. They are numerous. A single square centimetre of coral tissue can contain millions of algal cells. Despite being distinct organisms with their own DNA, the algae live entirely within the coral's body, protected from the outside environment, provided with carbon dioxide and nutrients from the coral's metabolism. These algae are often called zooxanthellae in everyday reef discussion. The term zooxanthellae is a bit of a catch-all, referring to several different species and groups of algae that form symbiotic relationships with corals and other marine organisms. The most common group belongs to the dinoflagellates, a type of single-celled algae known for their two whip-like flagella that they use to move when in a free-living stage. Inside the coral, however, the algae are non-motile, residing within the coral's cells, dividing and passing to new cells as the coral grows. The algae use sunlight to make energy through photosynthesis. Photosynthesis is the process by which light energy is converted into chemical energy, specifically sugars like glucose. The algae absorb sunlight, capture carbon dioxide from the surrounding tissue, and use water to build sugar molecules, releasing oxygen as a byproduct. This is the same fundamental process that drives plant growth on land, but here, it is happening inside an animal in the warm, shallow waters of a tropical reef. The oxygen released by the algae can be used by the coral, and the sugars produced can be shared. Some of that energy can be shared with the coral host. The algae don't keep all the sugar they produce. They release a portion of it, sometimes a substantial portion, into the coral's tissues. The coral absorbs this sugar and uses it as an energy source, fueling its metabolism, growth, and skeleton production. Estimates vary, but in some coral species, the algae can provide as much as 90% of the coral's energy needs. This means that the coral is, in a sense, partially solar-powered, relying on sunlight captured by its algal partners to fuel much of its daily activity. This partnership helps corals grow more efficiently in sunny, shallow water. Without the algae, corals would rely entirely on the food they can capture from the water, which in nutrient-poor tropical seas can be relatively scarce. The algae allow the coral to supplement its diet with photosynthetically produced energy, boosting growth rates and allowing the coral to invest more energy into skeleton production. This is why reef-building corals are most successful in clear, shallow waters where light is abundant. The deeper you go, the less light penetrates, and the less effective the algae become. Below a certain depth, corals can still survive by feeding, but the partnership with algae is less productive, and reef building slows or stops. The coral provides the algae with a safe place to live. Inside the coral tissue, the algae are protected from being eaten by planktivorous fish, or filtered out by other organisms. They are shielded from strong water flow that might sweep them away. They have a stable position in the water column, unlike free-floating plankton that drift wherever currents take them. This stability allows the algae to remain in the sunlit zone where photosynthesis is most efficient rather than being carried down into deeper, darker water. The coral also provides carbon dioxide and nutrients the algae can use. The coral's metabolism produces carbon dioxide as a waste product, which the algae can absorb and use in photosynthesis. The coral's digestion of captured food produces nitrogen and phosphorus compounds, which the algae need to build proteins and other essential molecules. In this way, the waste products of one partner become the raw materials for the other, creating a tight nutrient cycle that minimizes loss and maximizes efficiency. The reef, in a sense, recycles its nutrients internally, passing them back and forth between coral and algae, between animal and plant-like partner. The algae can give corals some of their colour. Corals come in many colours, from browns and greens to blues, purples, pinks and reds. Some of this colour comes from pigments in the coral tissue itself, but much of it comes from the algae. The golden-brown photosynthetic pigments of the algae give many corals their brown or greenish hue. When corals are stressed and expel their algae, a phenomenon known as bleaching, the coral tissue becomes translucent, revealing the white calcium carbonate skeleton beneath. This loss of colour is a visual indicator of the loss of the algal partnership and a warning sign that the coral is under stress. Because algae need light, many reef-building corals thrive in clearer waters. Turbidity, or cloudiness in the water, reduces light penetration. Sediment from rivers, runoff from land, or resuspension from storms can all increase turbidity, making it harder for the algae to photosynthesize effectively. Corals in murky water may receive less energy from their algae, forcing them to rely more on feeding, which can slow growth. This is one reason why reefs near river mouths, or in areas with heavy sedimentation, may be less well developed than reefs in clear oceanic waters far from land. This relationship is like a calm teamwork loop: shelter offered, energy shared, growth continued. The coral and algae don't negotiate. They don't communicate in any way we would recognize. The partnership is maintained by mutual benefit, by the simple fact that both organisms do better together than they would apart. The coral gains energy it couldn't easily obtain otherwise. The algae gain protection and nutrients. The reef grows because of this cooperation, layer upon layer, the result of millions of tiny partnerships playing out simultaneously across the reef's surface. Now we'll widen out to the reef environment itself, the conditions of warm water, light, and gentle currents that support reef life. Coral reefs don't form just anywhere in the ocean. They are found in specific regions where conditions are right, where the water is warm enough, clear enough, and shallow enough to support the coral-algae partnership. Understanding these environmental factors helps explain the global distribution of reefs and why they are so sensitive to changes in ocean conditions. Many coral reefs grow best in warm tropical and subtropical waters. If you were to draw the global distribution of coral reefs on a map, you would see them concentrated in a band around the equator, extending roughly 30 degrees north and south latitude. This corresponds to the warmest parts of the ocean, where surface temperatures rarely drop below 20 degrees Celsius and can reach 30 degrees or more in the summer. Temperature is critical because it affects the metabolic rates of both the coral and the algae. Warmer water within the tolerable range speeds up metabolism, allowing faster growth and more efficient energy use. Reefs are often found in shallow areas where sunlight can reach the bottom. Light intensity decreases exponentially with depth in the ocean. Even in the clearest water, only a fraction of surface light reaches depths beyond a few dozen meters. Most reef building occurs in water less than 30 meters deep, where light levels are high enough to support robust photosynthesis by the algae. Some corals can survive deeper, extending down to 50 or even 70 meters in very clear water, but these deeper corals grow more slowly and form less massive structures. The shallowest parts of the reef, just a few meters below the surface, often have the highest coral cover and the fastest growth rates, benefiting from maximum light exposure. Clear water helps sunlight travel deeper, supporting photosynthesis in reef algae. Water clarity is determined by the amount of dissolved and suspended material in the water. Sediments, plankton, dissolved organic matter, all of these scatter and absorb light. In clear oceanic water far from river inputs and with low plankton concentrations, light can penetrate deeply, supporting coral growth at greater depths. In turbid water near the coast or in areas with high runoff, light is quickly absorbed, and the productive reef zone is restricted to very shallow depths. Ocean currents can bring fresh nutrients and oxygen to reef areas. While the water around coral reefs is often low in dissolved nutrients like nitrogen and phosphorus, it is not entirely devoid of them. Currents flowing past the reef can carry plankton, organic particles, and dissolved nutrients that feed the reef community. Upwelling currents, which bring deeper, nutrient-rich water to the surface, can enhance productivity near reefs, though too much nutrient input can lead to problems like algal overgrowth. Oxygen is also important. Corals and other reef organisms need dissolved oxygen to respire, and water movement helps replenish oxygen that is consumed by the reef community. Currents also help carry away waste products from the reef. Just as a city needs a sewage system to remove waste, a reef benefits from water flow that flushes away metabolic byproducts, excess sediment, and other materials that might otherwise accumulate and harm the coral. Strong currents can bring challenges, such as increased physical stress or the risk of dislodging coral fragments, but moderate currents are generally beneficial, keeping the water fresh and well mixed. Waves can shape reef edges and influence where certain corals grow best. The reef crest, the shallowest part of the reef where waves break, is a high-energy environment. Only sturdy, wave-resistant corals can survive here, often forming low, robust shapes that can withstand the pounding surf. Below the crest, in the calmer waters of the lagoon or the forereef slope, more delicate branching or plate-like corals can thrive, taking advantage of the reduced wave energy to grow faster and more intricately. The physical environment shapes the coral community, and the coral community in turn shapes the physical environment, dissipating wave energy and protecting the shore. Some reefs form along coastlines while others form around islands. Fringing reefs grow directly adjacent to the shore, forming a narrow band of coral in the shallow water near land. These are the simplest type of reef in terms of structure, and are often found around volcanic islands and along tropical coasts. As you wade from shore into the water, you move from beach to shallow reef flat to the living reef, with deeper water beyond. Barrier reefs run parallel to shore, separated by a lagoon. These reefs are farther from the coast than fringing reefs, often several kilometres offshore, with a deeper lagoon of calm water between the reef and the land. The Great Barrier Reef is the most famous example, but barrier reefs exist in many parts of the tropics. The lagoon provides a sheltered environment for different communities of marine life, including seagrass beds, patch reefs, and sandy areas. The barrier reef itself takes the brunt of the ocean's wave energy, protecting the lagoon and the coastline beyond. Atolls are ring-shaped reefs that often form around sinking volcanic islands over long time. The formation of an atoll is a slow, geological process that can take millions of years. It begins with a volcanic island rising from the ocean floor. A fringing reef forms around the island's shore. Over time, the volcanic island begins to sink, a process driven by the cooling and subsidence of the oceanic crust. As the island sinks, the reef continues to grow upward, keeping pace with the rising relative sea level. Eventually, the island sinks completely below the surface, leaving only the reef, which now forms a ring around a central lagoon. Atolls are common in the Pacific and Indian Oceans, and they provide striking visual evidence of the interplay between geological and biological processes. Reefs are shaped by both biology and geography, like a city shaped by terrain and weather. The biology determines what organisms can live there, how they grow, how they interact. The geography determines the physical template, the slope of the seafloor, the exposure to waves, the proximity to land. Together, these factors create the unique character of each reef, making no two reefs exactly alike, even as they follow similar underlying principles. With the reef's neighbourhood established, we can stroll through the residents, the fish, invertebrates, and other life that finds calm shelter in reef spaces. The coral structure itself is only part of the story. The reef is also a habitat, a three-dimensional framework that provides living space for a vast array of organisms. The complexity of the reef structure creates niches, each one a potential home, feeding ground or nursery for some species. Coral reefs provide hiding places, feeding grounds, and nursery areas for many species. The crevices between coral branches, the overhangs beneath coral heads, the caves and tunnels that form in the reef structure, all of these offer shelter from predators. Small fish can dart into the coral when threatened, hiding in spaces too narrow for larger predators to follow. Juvenile fish often settle on reefs after a planktonic larval stage, finding refuge among the coral until they grow large enough to venture into more open water. The reef functions as a nursery for many commercially important fish species, supporting populations that extend far beyond the reef itself. Reef fish often use crevices and branching corals as shelter. Some fish are territorial, claiming a particular coral head or patch of reef as their own, defending it from intruders. Others move more freely, but still rely on the reef for refuge. At night, many fish settle into crevices to sleep, avoiding nocturnal predators. During the day, they emerge to feed, but they stay close to the reef, ready to retreat at the first sign of danger. The structural complexity of the reef is directly correlated with fish diversity and abundance. Reefs with more intricate structure support more species and more individuals than simpler, flatter habitats. Some fish graze on algae, helping keep surfaces from becoming overgrown. Herbivorous fish, such as parrotfish and surgeonfish, play an important role in maintaining the balance between coral and algae. Algae can grow quickly on reef surfaces, especially when nutrient levels are elevated. If left unchecked, algae can overgrow and smother coral, blocking light and preventing new coral larvae from settling. Herbivorous fish graze the algae down, cropping it close to the surface and preventing it from dominating the reef. This grazing is a form of ecosystem maintenance, a service provided by the fish that benefits the coral community. Sea urchins can also graze algae in some reef systems. These spiny invertebrates scrape algae from rock and coral surfaces using a specialized feeding structure called Aristotle's lantern, a complex arrangement of teeth and muscles. In some reefs, sea urchins are important herbivores, keeping algae in check. However, sea urchin populations can fluctuate dramatically. When predators are removed or when disease strikes, urchin populations can explode, sometimes grazing so intensely that they erode the reef surface itself. The balance between herbivores and algae, between grazing and growth, is delicate and can shift in response to changes in the ecosystem. Many invertebrates live on reefs, including shrimp, crabs, and sea stars. Invertebrates are animals without backbones, and they make up a large portion of reef biodiversity. Shrimp hide in coral branches and crevices, some of them forming cleaning stations where fish come to have parasites removed. Crabs scuttle across the reef, scavenging detritus and leftover food. Sea stars crawl slowly over the substrate, some feeding on coral, others on algae or detritus. Each species has its role, its niche, its way of making a living on the reef. Sponges are common reef residents and help filter water. Sponges are simple animals that pump water through their bodies, filtering out bacteria, plankton, and organic particles for food. A single sponge can filter thousands of litres of water per day, contributing to water clarity and nutrient cycling on the reef. Sponges come in many shapes and colours, from encrusting forms that coat the rock to large barrel sponges that can grow taller than a person. They are often overlooked compared to the more charismatic corals and fish, but they are an integral part of the reef community. Clams and other shellfish can live among corals and rocks. Giant clams, some of the largest mollusks in the world, nestle into crevices on the reef, their colourful mantles exposed to the sunlight. Like corals, giant clams have a symbiotic relationship with algae, hosting zooxanthellae in their tissues and benefiting from the energy the algae produce. Other bivalves burrow into the reef, some boring into the coral rock itself, creating holes that weaken the structure but also contribute to bioerosion, the biological breakdown of the reef that is part of the natural cycle of reef growth and decay. Some reef organisms are active during the day, while others come out at night. The reef has a day shift and night shift, two overlapping communities that use the same space at different times. Diurnal fish are active during the day, feeding on plankton, algae, or other fish. At dusk, they retreat into the reef to rest. As darkness falls, nocturnal animals emerge. Octopuses prowl the reef, hunting for crabs and shellfish. Moray eels slither out of their daytime hiding places. Basket stars unfurl their intricate arms to filter plankton from the water. The reef transforms at night. The cast of characters changes, but the underlying structure remains the same, providing a stage for both shifts. Reefs can be busy with life, but the daily rhythm often repeats gently. There is activity, yes, but it is not chaotic. Fish move in patterns, feeding at certain times, resting at others. Tides rise and fall, bringing slight changes in water depth and current. The sun rises and sets, driving the daily cycle of light and dark that structures the lives of so many reef organisms. There is a predictability to it, a rhythm that repeats day after day, year after year, a comforting constancy that is part of the reef's character. A reef community is like a quiet neighbourhood where many species share the same safe spaces. There is competition certainly for food, for space, for mates. There is predation, the endless cycle of hunter and hunted. But there is also cooperation, symbiosis, mutual benefit. There is coexistence, species living side by side, each exploiting a slightly different niche, each finding a way to make a living without excluding all others. The reef is crowded, but it is not a battlefield. It is a community, complex and interconnected, where the fortunes of one species can affect many others, where the whole is greater than the sum of its parts. Now we'll look at reef food webs, where energy moves through the system in calm loops rather than sharp stories. A food web is a map of who eats whom, a diagram of energy, flow through an ecosystem. On a coral reef, the food web is intricate with many pathways and connections. Energy enters the system primarily through photosynthesis, both by the zooxanthellae in corals and by free-living algae, and also through the capture of plankton from the water. From there, energy flows through grazers, predators, scavengers and decomposers, cycling and recycling until it is eventually lost as heat or exported from the reef. Reef food webs often begin with sunlight captured by algae and other photosynthetic organisms. The sun is the ultimate energy source for most life on Earth, and the reef is no exception. Photosynthetic organisms convert light energy into chemical energy, building sugars and other organic molecules that serve as fuel for themselves and for the animals that eat them. On a reef, this primary production happens both within the coral tissues, via the zooxanthellae, and on the reef surfaces where turf algae, coralline algae and other photosynthetic organisms grow. Coral-hosted algae contribute energy that supports coral growth. As we've discussed, the zooxanthellae provide much of the energy that reef-building corals use to grow and build their skeletons. This energy doesn't just stay within the coral. When coral polyps produce mucus, some of that mucus is released into the water, where it can be consumed by other organisms. When coral tissue is damaged or grazed, that organic material enters the food web. The energy captured by the zooxanthellae ripples outward, supporting not just the coral but also the wider community. Plankton in the water also provides food for many reef animals. Plankton includes both phytoplankton, which are photosynthetic, and zooplankton, which are animals or animal larvae. Plankton drifts with the currents, and reefs, with their complex structure, can act as plankton traps, capturing drifting organisms from the water. Corals feed on plankton, as do many other reef animals. Filter feeders, like sponges, clams, and feather stars, capture plankton from the water column. Planktivorous fish, such as damselfish and fusiliers, pick individual plankton items from the water as they drift past. Small fish may feed on plankton drifting past the reef. These planktivorous fish often form schools, hovering above the reef in the current, picking off zooplankton as it flows by. The fish position themselves where the current brings a steady supply of food, reducing the energy they need to spend searching. This feeding strategy is efficient, but it also exposes the fish to predators. Schooling provides some safety in numbers, and the proximity to the reef allows for quick retreat when danger threatens. Herbivorous fish feed on algae and help recycle nutrients. When a parrotfish scrapes algae from a rock, it also ingests some of the rock itself, grinding it up with powerful teeth and a beak-like jaw. The fish digests the algae, extracting nutrients, and excretes the inorganic material as fine sand. This process contributes to the production of beach sand on tropical islands. The nutrients in the algae are absorbed by the fish, fueling its growth and metabolism, and eventually return to the reef through the fish's waste or when the fish itself is eaten or dies. Predatory fish may feed on smaller fish, balancing populations over time. Predators like groupers, snappers, and sharks occupy the upper levels of the reef food web. They help control the populations of prey fish, preventing any one species from becoming too abundant. This predation pressure can influence prey behavior, making prey fish more cautious, more likely to stay close to shelter, more vigilant. The presence of predators shapes the structure of the prey community, a phenomenon known as top-down control. Many reef creatures are scavengers that clean up leftover material. When a fish dies, its body doesn't go to waste. Scavengers like crabs, shrimp, and small fish quickly find the carcass and consume it, recycling the nutrients back into the living community. Detritus, the bits and pieces of organic material that settle on the reef, is consumed by detritivores. Animals specialise for feeding on this material. Nothing is wasted on the reef. Energy and nutrients are cycled and recycled, passed from organism to organism until nearly all the value has been extracted. Decomposers like bacteria help break down organic matter into nutrients. Bacteria are microscopic, but they are present in vast numbers on the reef, coating surfaces, living in sediment, floating in the water. They break down complex organic molecules into simpler forms, releasing nutrients like nitrogen and phosphorus that can be taken up by algae and other photosynthetic organisms. This decomposition closes the nutrient loop, returning elements to forms that can be used again by primary producers, keeping the system productive even in nutrient-poor waters. This recycling helps the reef remain productive even in nutrient-poor tropical waters. The tropical oceans are often described as biological deserts, places where the surface waters are clear and blue precisely because they lack the nutrients that would support large blooms of plankton. And yet, reefs thrive in these waters. The key is efficiency. Reefs are very good at capturing and retaining nutrients, passing them from organism to organism, recycling them through the food web, minimizing loss to the surrounding ocean. The reef is a nutrient oasis in a nutrient desert, maintaining high productivity through tight internal cycling. The reef's energy system is like a slow, steady circle, sun to life to nutrients and back again. Energy flows in one direction, from the sun through photosynthesis to herbivores to predators, eventually dissipating as heat. But nutrients cycle, moving from organism to organism, from living tissue to waste to decomposition to uptake, around and around. The reef is powered by sunlight, but sustained by recycling, a closed-loop system that wastes little and uses much. With those gentle cycles in place, we can drift into coral growth itself, how reefs build their layers, slowly and steadily. The physical structure of the reef, the massive accumulations of limestone that form islands and atolls, is the product of countless coral polyps secreting calcium carbonate over long periods of time. Understanding how this process works, at the level of individual polyps and at the level of entire reef systems, helps us appreciate the scale and patience of reef building. Reef-building corals deposit calcium carbonate to create their hard skeletons. The process is called calcification, and it requires energy. The coral polyp takes calcium ions and carbonate ions from the seawater and combines them to form calcium carbonate, which is deposited as aragonite, a crystalline form of calcium carbonate, at the base of the polyp. This deposition is controlled by the coral cells and occurs at specific sites where the skeleton is being built. The exact mechanisms are still being studied, but it is clear that the process is linked to the coral's metabolism and is enhanced by the presence of zooxanthellae. The skeleton acts like a foundation that stays after the polyp moves or dies. The calcium carbonate skeleton is permanent in the sense that it doesn't dissolve or decay quickly. When a polyp dies, its skeleton remains, a hard structure that can persist for years, decades, centuries. New polyps can settle on old skeletons, or existing polyps can grow over them, incorporating them into the living reef. The reef, therefore, is a structure where the past supports the present, where the accumulated skeletons of former generations provide the foundation for the living tissue of today. New coral polyps can grow on older skeleton layers, adding height and complexity. As a coral colony grows, it leaves behind a trail of skeleton. Branching corals add new branches at their tips, leaving a skeletal trunk behind. Massive corals add layers like tree rings, expanding outward and upward. The colony grows taller, broader, heavier, all from the incremental addition of calcium carbonate by individual polyps. Over time, these incremental additions create structures that can be meters tall, meters across, weighing many tons. Branching corals can create intricate structures that shelter many small animals. The branches form a three-dimensional lattice, full of spaces and gaps that small fish, shrimp, crabs, and other animals can inhabit. These structures are fragile compared to massive corals, but they grow faster and provide more surface area and more hiding places per unit volume. In calm, protected waters, branching corals can dominate, creating thickets that look like underwater forests, swaying gently in the current, alive with movement and colour. Massive corals grow more slowly, but can become large and sturdy over time. These corals form mounds or boulders, growing outward in all directions at a slow, steady pace. A massive coral head might grow only a few millimeters per year, but it can live for centuries, eventually becoming a structure several meters across. These corals are more resistant to storm damage than branching corals, and they often dominate in high-energy environments where waves and currents are strong. Their slow growth is a trade-off for durability, a strategy that favours long-term persistence over rapid expansion. Reef growth often happens in layers, like slow stonework built by tiny hands. If you could cut through a coral head and examine its interior, you would see growth bands, layers of denser and lighter skeleton that correspond to seasonal or annual variations in growth rate. These bands are similar to tree rings and can be used to determine the age of the coral and to study past environmental conditions. The layers tell a story of the coral's life, recording years of steady growth, years of stress, years of recovery. Broken coral. Pieces can sometimes become new growth sites if they settle in suitable places. Coral fragments broken off by storms or other disturbances don't necessarily die. If a fragment lands in a suitable location, where it won't be buried by sediment or swept away by currents, it can survive, attach to the substrate, and begin growing again. This process, called fragmentation, is a form of asexual reproduction that allows corals to colonize new areas and recover from damage. Some corals are more prone to fragmentation than others, and in some reef systems, fragmentation is an important means of reef expansion. Over long time spans, reefs can grow into large formations visible from above. The Great Barrier Reef, as mentioned, is visible from space. Other reefs form entire archipelagos of islands, rings of coral sand and rock enclosing lagoons. From the air, these formations are breathtaking, complex patterns of blue and green and white, the product of biological processes operating over geological timescales. The reef's growth is imperceptible from day to day, but given centuries or millennia, it can reshape coastlines, create islands, alter ocean currents. Reefs can also record environmental history in their layers, like a quiet diary made of limestone. Scientists can drill cores from massive corals, extracting columns of skeleton that span decades or centuries. By analysing the chemical composition of the skeleton, they can reconstruct past ocean temperatures, salinity, and chemistry. The skeleton incorporates trace elements and isotopes from the seawater in which it formed, and these chemical signatures vary with environmental conditions. The coral becomes an archive, a record of the ocean's past, written in calcium carbonate and read by scientists seeking to understand climate change and ocean history. The main message of reef building is patience, small additions becoming something vast. There is no shortcut to building a reef. It cannot be rushed. It requires time, stability, and the persistent effort of countless polyps doing what polyps do, day after day, year after year. The reef is a monument to slow, steady work, a structure that embodies patience in physical form. In a world that often prizes speed and immediate results, the reef offers a different lesson, a reminder that some of the most enduring and impressive things are built incrementally, patiently, without fanfare. Now we'll gently explore the daily coral routine: feeding, resting, and responding to light in a calm rhythm. Corals are not static. They respond to their environment, adjusting their behavior in subtle ways to maximize food capture, minimize damage, and maintain their symbiotic relationship with algae. The daily cycle of light and dark drives much of this behavior, creating a rhythm that repeats with comforting regularity. Many corals extend their tentacles more at night to catch plankton. During the day, with the sun overhead and the algae photosynthesizing, many corals keep their tentacles retracted. This reduces the risk of damage from grazing fish and minimizes energy expenditure. But as dusk approaches and light levels drop, the polyps begin to extend their tentacles, unfurling them into the water. By full dark, the reef surface that looked smooth and closed during the day is now fuzzy with tentacles, each polyp reaching out to capture food from the water. This transformation happens gradually, a slow blooming as the reef shifts from day mode to night mode. During the day, corals often rely more on energy from their algae partners. The zooxanthellae are hard at work during daylight hours, capturing sunlight and producing sugars. The coral benefits from this energy, using it to fuel metabolism and growth. In some sense, the coral can rest during the day, relying on its internal solar panels to provide energy, and save active feeding for the night when plankton is more abundant and the competition from diurnal planktivores is reduced. Corals can sense light and may adjust behaviour based on day and night cycles. Corals lack eyes, but they have photoreceptors, light-sensitive proteins that can detect the presence and intensity of light. These photoreceptors help the coral coordinate its behaviour with the daily light cycle, timing tentacle extension, spawning, and other activities to occur at optimal times. The ability to sense light also helps the coral optimize the positioning of its zooxanthellae, ensuring they receive adequate sunlight for photosynthesis. Coral polyps can retract tentacles for protection. When disturbed by a fish brushing against them, by a diver's hand, by a strong current, coral polyps can quickly pull their tentacles inward, drawing them into the safety of the coral light. This reflex protects the delicate tentacles from damage. Once the disturbance passes, the tentacles slowly re-extend, resuming feeding or simply remaining in their retracted state until conditions are favourable again. Mucus, produced by corals, can help remove sediment from their surfaces. Corals secrete a layer of mucus that covers their tissue. This mucus serves several functions. It can trap sediment, which is then sloughed off, carrying the sediment away from the polyp. It can contain antibacterial compounds that protect against infection. It can provide a surface on which beneficial bacteria live. The mucus layer is constantly being produced and shed, a form of self-cleaning that helps the coral maintain a clean surface for gas exchange and feeding. Some reef fish and invertebrates help keep coral surfaces cleaner by grazing nearby. Herbivorous fish that feed on algae often graze right up to the edge of coral colonies, removing algae that might otherwise encroach on the coral. Some small invertebrates, like certain crabs and shrimp, live among coral branches and pick off debris, parasites, or competing organisms. These cleaning services are beneficial to the coral, helping it maintain health and avoid being overgrown. Water movement helps bring food and oxygen to coral polyps. Currents and wave action keep the water around the reef well mixed, preventing stagnant zones where oxygen could be depleted or waste products could accumulate. The movement of water past the polyps ensures a constant supply of fresh plankton and dissolved oxygen, while carrying away carbon dioxide and other metabolic waste. Corals in areas with good water flow often grow better than those in stagnant conditions, all else being equal. Corals also release waste products that are carried away by currents. Like all animals, corals produce waste from metabolism and digestion. Ammonia and other nitrogenous wastes are excreted into the water, where they are diluted by currents and taken up by algae and bacteria. The efficient removal of waste is important for preventing the buildup of toxic substances that could harm the coral or its algal symbionts. The reef's daily rhythm repeats gently, like a slow tide of activity and quiet. Day after day, the sun rises, the algae photosynthesize, the polyps rest. Night after night, darkness falls, the polyps extend their tentacles, plankton is captured. The rhythm is predictable, comforting in its regularity. There are variations, of course, storms that disrupt the pattern, seasonal changes in temperature or plankton abundance, but the underlying cycle remains, the heartbeat of the reef, steady and enduring. In many ways, corals live by small routines that add up to long-term success. They don't innovate. They don't change strategies based on short-term conditions. They do what they have evolved to do, day after day, year after year, and the cumulative effect of this consistency is a thriving reef. There is something reassuring in this, the idea that success can come from simply doing the right small things, repeatedly, patiently, without seeking shortcuts or quick wins. As we near the end, we can look at how people study reefs and why these slow-growing cities are treated with such careful respect. Coral reefs are not just natural wonders, they're also systems of immense ecological and economic value. They provide food, coastal protection, and livelihoods for millions of people. They are centres of biodiversity, home to species found nowhere else on Earth, and they are vulnerable, sensitive to changes in temperature, water quality, and other environmental factors. Understanding reefs, monitoring their health, and protecting them for the future are goals that drive much of marine science. Marine scientists study reefs using diving surveys, cameras, and underwater mapping tools. Researchers put on scuba gear and descend to the reef, swimming transects, taking notes, identifying species, measuring coral cover and fish abundance. Underwater cameras capture images and video that can be analysed later, providing a permanent record of the reef's condition. Mapping tools, including sonar and photogrammetry, create detailed three-dimensional models of reef structure, allowing scientists to track changes in reef shape and size over time. Some reefs are monitored over years to track changes in growth and species presence. Long-term monitoring programs return to the same sites year after year, documenting how the reef changes. This long-term perspective is essential for understanding reefs, which change slowly under normal conditions and can take years to recover from disturbances. By comparing data from different years, scientists can detect trends, identify stressors, and assess the effectiveness of conservation measures. Scientists measure coral cover, fish populations, and water conditions to understand reef health. Coral cover, the percentage of the reef surface covered by living coral, is a common metric of reef health. Declines in coral cover can indicate stress from bleaching, disease, or physical damage. Fish populations provide information about the reef's ecological function and the integrity of the food web. Water conditions, including temperature, salinity, nutrient concentrations, and turbidity, help explain why reefs are healthy or stressed. Coral skeletons can be studied to learn about past ocean temperatures and chemistry. As mentioned earlier, coral cores provide a record of environmental conditions extending back decades or centuries. By analysing the ratios of different oxygen isotopes in the skeleton, scientists can estimate past sea surface temperatures. By measuring the incorporation of trace elements like strontium, they can infer changes in ocean chemistry. These proxy records help scientists understand natural climate variability and put recent changes into a long-term context. Reef biodiversity is often compared across regions to see patterns in ecosystems. Reefs in different parts of the world have different assemblages of species, shaped by geography, history, and local environmental conditions. Comparing reefs in the Caribbean to reefs in the Pacific, for example, reveals differences in species richness, dominant coral types, and ecological dynamics. These comparisons help scientists understand the factors that control biodiversity and predict how reefs might respond to future changes. Some reefs are protected areas where fishing and human impact are limited. Marine protected areas, or MPAs, are regions where human activities are restricted to conserve the ecosystem. In some MPAs, all fishing is banned. In others, only certain types of fishing are allowed. Protected reefs often have higher coral cover, more fish, and greater biodiversity than unprotected reefs, demonstrating the benefits of conservation. These areas also serve as reference sites, showing what reefs can look like in the absence of heavy human pressure. Reef restoration efforts can include growing corals in nurseries and transplanting them carefully. When reefs are damaged, either by storms, bleaching, or human activities, active restoration can help speed recovery. Coral fragments are collected and grown in underwater nurseries, structures where they can grow without being disturbed by predators or buried by sediment. Once the fragments have grown large enough, they are transplanted back onto the reef, where they can continue to grow and eventually reproduce. Restoration is labour-intensive and cannot replace the lost complexity of a mature reef, but it can help jumpstart recovery and maintain coral populations. Research on reefs often emphasises patience, because reefs change slowly. Unlike some ecosystems where change can be rapid and dramatic, reefs operate on a timescale that can frustrate researchers accustomed to quick results. A study of reef growth might require decades of data. A restoration project might not show significant success for years. But this slow pace is part of what makes reefs reefs. Understanding them requires patience, a willingness to observe carefully over long periods, to trust in slow processes, to value subtle changes. Many reef questions are answered through long-term observation rather than quick experiments. While experiments have their place in reef science, much of what we know about reefs comes from simply watching, from returning to the same sites again and again, from documenting what is there and how it changes. This observational approach is well suited to systems that change slowly and where interventions might have unintended consequences. It is a humble approach, one that acknowledges that we are still learning, that reefs are complex and not fully understood. The way reefs are studied matches their nature, calm, careful, and steady over time. There is a harmony between the subject and the method, between the reef's slow growth and the patient observation of scientists who dedicate careers to understanding these systems. The work is not flashy. It involves long hours underwater, careful data entry, meticulous analysis. but it is important work that contributes to the conservation of some of the most valuable ecosystems on the planet. Now we'll bring the reef back to bedtime scale, considering slow growth, gentle teamwork, and the soothing comfort of steady patterns. Coral reefs, for all their complexity and diversity, can also be a source of calm contemplation. They embody principles that resonate with a restful mindset: patience, cooperation, incremental progress, rhythmic cycles. Thinking about reefs in this way can be a way to slow the mind, to step away from the hurried pace of daily life, and to find peace in the idea of slow, steady growth. Coral reefs are built by tiny animals doing small actions repeatedly. Each polyp is small, its contribution to the reef is small, its daily activities are simple and repetitive. But multiply that small contribution by millions of polyps, by thousands of years, and you get a structure of immense size and complexity. The reef is proof that small actions matter, that consistency over time can achieve what no single burst of effort could accomplish. Their beauty comes from accumulation, not sudden change. A reef is not built in a day. It is not the product of a single dramatic event. It is the result of countless small additions, each one insignificant on its own, but together creating something magnificent. This principle applies beyond reefs. Many worthwhile things in life are built slowly, through daily effort, through patience and persistence. The reef is a reminder of this, a physical embodiment of the power of accumulation. The reef structure creates shelter, like a home with many rooms. The reef is a place of refuge, a haven for countless species. Its complexity provides niches, each one a safe space for some organism. This idea of shelter, of a safe place built by community effort, is comforting. The reef is not just a structure, it is a home, a community, a place where life can flourish because the environment supports it. Reef life follows routines, light and dark, feeding and resting, currents and calm. There is a rhythm to reef life, a predictable cycle that repeats day after day. This rhythm is stabilizing. It allows organisms to coordinate their activities, to time feeding, reproduction, and rest to occur when conditions are optimal. The rhythm is also soothing, a reminder that there is order in nature, that cycles repeat, that after dark comes light, after activity comes rest. The coral-algae partnership shows how cooperation can create stability. The symbiosis between coral and zooxanthellae is a relationship built on mutual benefit, where each partner contributes to the success of the other. This cooperation creates a system that is more productive, more resilient, more stable than either partner could be alone. It is a model of how working together can achieve more than working apart, a lesson that applies to human societies as much as to coral reefs. Reefs remind us that strong systems can be quiet systems. The reef does not announce its strength with noise or aggression. It sits quietly beneath the waves, enduring storms, supporting life, growing incrementally. Its strength comes from its structure, from the accumulated efforts of countless polyps, from the tight cycling of nutrients, from the cooperation between coral and algae. Strong does not have to mean loud or fast. It can mean steady, resilient, patient. Thinking about reef growth can slow the mind, like watching sand settle in clear water. There is something meditative about contemplating processes that occur on timescales far longer than our own daily experience. Imagining a coral polyp adding a thin layer of skeleton day after day, year after year, can put our own hurried activities into perspective. It can remind us that not everything needs to be rushed, that some things are worth waiting for, that patience is not passive but an active choice to trust in slow processes. Tonight, you don't have to be fast, you only have to be gentle. Just as a coral polyp doesn't rush, doesn't force growth, doesn't skip steps, you can allow yourself to rest without urgency. There is no deadline for falling asleep, no requirement to achieve anything before morning. You can simply be, like a polyp resting in its coral light, breathing slowly, existing in the quiet rhythm of night. Like a reef, rest is built in small layers, one exhale then another. Sleep doesn't come all at once. It arrives gradually in stages, deepening with each breath, each moment of stillness. You settle into it the way sediment settles onto the reef, slowly drifting down, finding a place to rest. There is no force required, No effort, just a gentle letting go, a trust in the process, an acceptance that rest will come in its own time. And as you fall asleep, you can imagine your thoughts becoming a calm reef, steady, safe, and softly glowing. Picture the reef in your mind, the gentle sway of coral in the current, the soft colours muted in the dim light, the quiet presence of fish resting in, crevices, the slow pulse of the ocean overhead. Let your thoughts slow to match that rhythm. Let the complexity of the day settle into the simple pattern of breath and heartbeat. The reef is there, timeless and patient, and you can rest within that image, safe and calm. Take a slow inhale, drawing breath like the reef draws water, gently, without effort. And then a longer exhale, releasing tension, releasing thought, letting everything flow out like the tide pulling away from shore. And let your mind settle like warm water over a quiet reef, smooth and unhurried, finding its level, becoming still. The reef continues its slow work beneath the waves, and you continue your slow descent into sleep. Both processes natural, both processes gentle, both processes complete in their own time. The next story is about to begin. Please relax and enjoy. Honeybees are social insects that live together in organized colonies, and there's something deeply calming about that simple fact. These small creatures, found on every continent except Antarctica, have evolved over millions of years to work as a collective rather than as individuals. When we say they're social, we mean something more than just living near each other. Honeybees coordinate their activities, share resources, and divide labour in ways that allow the colony to function as a single breathing organism. The colony itself is often called a hive, though technically the hive refers to the structure they live in, whether it's a hollow tree, a wooden box provided by a beekeeper, or a cavity in a wall. But the word has come to mean both the home and the family inside it, and that's fitting, because for honeybees, the two are inseparable. Honeybees are best known for two things: making honey and helping pollinate flowering plants. Both of these activities have shaped human agriculture and natural ecosystems for thousands of years. Pollination happens when pollen, the fine powder produced by a flower's male parts, is moved to the female parts of another flower of the same species, helping plants make seeds and reproduce. Honeybees accomplish this almost accidentally as they visit flowers searching for food. Their bodies become dusted with pollen, and as they move from bloom to bloom, some of that pollen rubs off in just the right place. It's a gentle exchange, one that happens millions of times each day during the growing season, and it's essential for many of the crops humans depend on, and for wildflowers that sustain entire ecosystems. But honeybees are just one of many pollinators working quietly in fields and gardens around the world. Butterflies drift from flower to flower on warm afternoons. Moths visit night-blooming plants under moonlight. Hummingbirds hover with astonishing precision, their long beaks reaching deep into tubular flowers. Other species of bees, bumblebees, mason bees, leaf-cutter bees, each have their own strategies and preferences. Even some beetles, flies, and bats contribute to pollination in different parts of the world. The honeybee's role is significant, but it exists within this larger tapestry of pollinating creatures, each playing a part in keeping flowering plants reproducing season after season. A honeybee colony typically includes 3 types of individuals: a queen, many workers, and, at certain times of the year, drones. Each type has a distinct role, and together they form a system that can persist for years if conditions remain favourable. The colony's population can number anywhere from 10,000 to 60,000 bees or more, depending on the season and the health of the hive. All these individuals are bound together by shared genetics, shared labour, and shared chemical signals that flow through the hive like invisible threads. Honeybees are insects, which means they belong to a group of animals characterised by having 6 legs and a body divided into 3 main parts. If you were to look closely at a honeybee resting on a flower, you'd see its head at the front with large compound eyes and delicate antennae. Behind the head is the thorax, the middle section where the legs and wings attach. And finally there's the abdomen, the segmented rear portion that houses much of the bee's digestive system, and, in workers and queens, the sting. This 3-part body plan is shared by all insects, from beetles to butterflies to ants, and it's proven remarkably successful over hundreds of millions of years of evolution. The antennae deserve special mention because they are incredibly important to a honeybee's experience of the world. Each antenna is covered with thousands of tiny sensory structures that detect chemicals in the air, allowing the bee to smell flowers from a distance, recognize nest mates, and perceive the pheromones that carry information through the hive. The antennae also sense vibrations and can detect air currents, helping the bee navigate and communicate. In many ways, a honeybee experiences reality more through scent and touch than through sight, even though its eyes are quite capable. A calm way to picture a hive is like a small town with shared work and shared warmth. There's no mayor, no city council, no central planner dictating what needs to be done. Instead, each bee responds to local conditions, what it senses around it, what signals it receives from other bees, what tasks seem most urgent based on the hive's current needs. And somehow, from thousands of individual decisions made moment by moment, order emerges. Food gets stored, young get fed, the temperature stays regulated, and the colony persists. It's a kind of distributed intelligence, a system where the whole is far greater than the sum of its parts, and there's something deeply soothing about that idea as you settle into sleep. Now that we've met the bees themselves, we can drift into the hive's gentle structure, how a colony stays organized without rushing, how the physical space of the hive is built and maintained through patient, repetitive work. Honeybees build honeycomb from wax they produce in their own bodies, which is remarkable when you think about it. Worker bees have special glands on the underside of their abdomen that secrete tiny flakes of wax. These flakes are initially clear and soft, almost like small scales. A bee will use its legs to move a wax scale from its abdomen to its mouth, where it's chewed and mixed with saliva to make it more pliable. Then, with careful, deliberate movements, the bee adds this softened wax to the growing comb, shaping it cell by cell. The process is slow and methodical, the kind of work that requires no hurry, just steady attention. Honeycomb cells are often hexagonal, forming a pattern of six-sided chambers that fit together perfectly with no gaps. This shape is mathematically efficient, uses less wax to enclose a given amount of space than other shapes would, and it provides structural strength. Bees don't understand hexagonal geometry in an abstract sense, of course, but through their building process, guided by instinct and the constraints of physics, hexagons naturally emerge. The walls of each cell tilt slightly upward, which helps prevent liquid honey from dripping out. The overall effect is a beautiful repeating pattern that's both functional and aesthetically pleasing, like a natural work of architecture that grows organically from the bees' collective effort. The honeycomb serves multiple purposes. It's where honey is stored after being processed from nectar. It's where pollen is packed away as a protein reserve. And it's where the next generation of bees develops, with eggs laid in individual cells that become nurseries for larvae and pupae. This multi-use approach makes the honeycomb incredibly efficient. The same basic structure, built from the same basic material, can serve as pantry, nursery, and even structural support for the hive. And because wax is produced by the bees themselves from the honey they eat, the comb is entirely renewable, a closed loop of resources that the colony manages internally. The inside of a hive is usually kept warm and steady, typically around 93 to 95 degrees Fahrenheit in the area where young bees are developing. This is warmer than most people keep their homes, and maintaining it requires constant effort, especially when the outside temperature drops. Bees have evolved several strategies for temperature regulation that don't require any technology, just cooperation and the collective heat of many small bodies working together. When it's cool outside, bees can cluster together to conserve heat. They pack tightly into a ball-shaped mass, with bees on the outside forming an insulating shell and bees on the inside staying warmer. The cluster isn't static. Bees slowly rotate positions, with colder bees from the outside gradually moving inward and warmer bees moving outward, ensuring that no individual bee gets too cold. This rotation happens continuously, a slow circulation that distributes warmth throughout the cluster. Inside the cluster, bees vibrate their flight muscles without actually flying, generating heat through muscle activity. It's the same principle as shivering in humans, but much more controlled and efficient. When it's hot, the challenge reverses. Bees can help cool the hive by fanning their wings, creating air currents that move heat out of the hive and bring cooler air in. If you've ever stood near a beehive entrance on a hot day, you might feel a faint breeze. That's the collective effort of many bees fanning in coordination. Some bees position themselves at the entrance and fan air out, while others might station themselves deeper inside to keep air circulating through the combs. It's organised ventilation achieved without any mechanical fans or electricity, just the coordinated beating of thousands of wings. Bees may also bring water into the hive to help with cooling through evaporation. Forager bees collect water from puddles, streams, or droplets on leaves, carry it back in their honey stomachs, and distribute it throughout the hive. Worker bees spread thin films of water on the comb, and as the water evaporates, it absorbs heat from the surrounding air, cooling the hive much like sweat cools a human body. On very hot days, you might see bees making many trips to water sources, carrying back tiny amounts that collectively make a significant difference in the hive's internal climate. The hive is dark inside, nearly pitch black in the deep areas away from the entrance. Very little light penetrates through the layers of comb and the massive bee bodies, so bees rely heavily on smell and touch to navigate their home. Their antennae constantly brush against surfaces, sensing the texture of wax, the presence of other bees, and the chemical signatures that mark different areas of the hive. Smell provides a rich tapestry of information: the sweet scent of honey, the protein-rich smell of stored pollen, the distinctive odours of developing larvae, and the pheromones of the queen that permeate the colony. In this dark, fragrant world, bees move with confidence, their senses perfectly adapted to the environment they've created. A well-run hive can feel like a quiet, living building made of teamwork. Every cell of honeycomb represents hours of wax production and careful shaping. Every gram of stored honey represents multiple foraging trips, and the collective effort of many bees processing nectar. The stable temperature represents constant vigilance, and the willingness of thousands of individuals to contribute their warmth or their wing beats to the common good. There's no fanfare, no celebration of achievement, just the steady hum of work continuing day after day, and that steady hum is the sound of success in the honeybee world. With the home in place, let's gently meet the three main roles in the colony: queen, workers, and drones, and understand how each contributes to the hive's continuation. The queen is the main egg layer in a honeybee colony, and in most cases, she's the only individual laying eggs. Her role is absolutely central to the colony's survival, because without a constant supply of new bees, the population would gradually decline as older workers reach the end of their natural lifespan. A healthy queen can lay up to 1,500 to 2,000 eggs per day during peak season, working her way methodically through the comb, pausing at each empty cell to inspect it, and, if suitable, deposit a single egg. This is exhausting work, and the queen receives constant attention from worker bees who feed her, groom her, and clear a path for her as she moves across the comb. The queen is usually larger than worker bees in body length, though not dramatically so. Her abdomen is notably longer and more tapered, which makes sense given that it houses her enlarged reproductive organs. Her wings appear shorter relative to her body, though this is more an optical illusion created by her longer abdomen than an actual difference in wing size. If you were to see a queen on a comb surrounded by workers, you'd likely notice her immediately, not just because of her size, but because of the circle of workers usually attending her, all oriented toward her, constantly touching her with their antennae and mouth parts. The queen produces chemical signals called pheromones that help organize colony behavior. The most important of these is queen substance, a complex blend of chemicals that she secretes from glands in her head. Workers who come into contact with the queen pick up these pheromones and spread them throughout the hive by touching other bees and walking across the comb. This creates a chemical presence that permeates the entire colony, signaling that the queen is alive, healthy, and actively laying eggs. As long as this signal is strong, workers remain focused on their normal tasks. If the signal weakens, because the queen has died, become injured, or is simply getting old, workers begin preparations to raise a new queen, demonstrating how chemical communication can coordinate major colony-level decisions. Worker bees are female bees that do most of the colony's tasks. They're the foragers you see visiting flowers, the builders constructing comb, the nurses feeding larvae, the guards defending the entrance, and the undertakers removing dead bees from the hive. Workers make up the vast majority of the colony's population, and though they're anatomically capable of laying eggs, they normally don't. The queen's pheromones suppress the workers' reproductive systems, keeping them focused on supporting the colony rather than reproducing themselves. In a sense, Workers are the queen's daughters who've sacrificed their own reproduction to help raise their sisters, a strategy that makes sense from an evolutionary perspective because of the unusual way genetics work in honeybees. Workers clean cells, preparing them for new eggs or for honey storage. They feed young larvae, visiting each larva multiple times per day with nutritious secretions. They build wax comb, shaping it into the precise hexagonal cells that will hold the colony's resources. They forage for food, flying out to find flowers, collect nectar and pollen, and navigate back home. They guard the hive entrance, checking incoming bees to ensure they belong to the colony and not to a neighbouring hive intent on robbing stored honey. They regulate temperature, either clustering for warmth or fanning for cooling. They process nectar into honey, standing over open cells and fanning their wings to evaporate excess water. They remove debris, dragging out dead bees, wax scraps, and anything else that doesn't belong in the hive. This list could continue, but the point is clear. Workers do essentially everything except lay the eggs that become the next generation. Drones are male bees whose main role is to mate with a queen from another colony. This is their sole reproductive purpose, and their anatomy reflects this specialization. Drones are larger and stockier than workers, with big eyes that meet at the top of their head, giving them excellent vision for spotting queens during mating flights. They have no pollen baskets, no wax glands, and no stinger. They're built for flying and mating, not for the various tasks that occupy workers. Drones do not collect pollen or build comb. They're fed by worker bees and spend much of their time resting on the comb or taking short orientation flights to familiarise themselves with the area around the hive. The number of drones in a hive changes with the season. In spring and early summer, when new colonies might be forming and young queens taking mating flights, the colony raises hundreds or even thousands of drones. Resources are plentiful, and the chance of successful mating is worth the investment. But as summer turns to fall and flowers become scarce, drones are seen as a liability. They consume honey, but contribute nothing to the colony's winter survival. Worker bees begin to drive drones out of the hive, blocking them from returning and even pulling them out of cells if they try to hide. It sounds harsh, but it's a practical response to limited resources. Drones cannot survive on their own, and by late fall, most have died. The colony will raise new drones again the following spring when conditions improve. A colony's strength depends on many factors, including food supply, the health of the queen, the absence of diseases and pests, and favourable weather. A strong colony in good conditions can grow rapidly, filling box after box with comb and honey if managed by a beekeeper, or preparing to swarm if left to their own devices. A weak colony might struggle to maintain its population, falling behind in building comb or storing food, and becoming vulnerable to robbing by stronger colonies. The factors affecting colony health are interconnected. A failing queen leads to fewer young bees being raised, which leads to fewer foragers, which leads to less food collected, which weakens the remaining bees, and so on in a downward spiral. Conversely, a healthy queen, abundant flowers, and favourable weather can create a virtuous cycle of growth and prosperity. The colony works best when each role fits into the larger rhythm of the hive. Queens lay eggs, workers perform the tasks needed to support the colony, and drones await their chance to mate. No single bee understands the whole system, but each responds to the conditions around it, and from those local interactions, colony-level patterns emerge. It's a kind of bottom-up organisation, where order arises from simple rules, followed by many individuals rather than from top-down commands, and this decentralised approach has proven remarkably resilient, allowing honeybee colonies to persist for millions of years across changing climates, evolving predators, and shifting floral landscapes. Now we'll follow the gentle timeline of a worker bee's life, how jobs change with age, like a quiet routine that evolves naturally, without planning or promotion, just a gradual shift from interior work to outside foraging. Worker bees often start with tasks inside the hive when they are young, typically within the first day or two after emerging from their cells as adults. These early tasks can include cleaning cells to prepare them for new eggs or for honey storage. A newly emerged worker might spend hours inspecting cells, removing any debris and polishing the cell walls until they're smooth and ready. This work doesn't require extensive experience or training. It's simple, repetitive, and important. Every cell that will house a developing bee or store food needs to be clean, and young bees provide that service while they're still orienting themselves to hive life. Workers may also help keep the hive warm by joining the cluster, especially if they emerge during a cool period. Even young bees can generate heat through muscle activity, and by positioning themselves within the cluster, they contribute to the collective warmth that protects developing larvae and pupae. This is passive work in a sense. They don't need to do anything except be present and maintain their body temperature, but it's crucial for the colony's brood-rearing success. As workers age, they often move into caring for larvae, sometimes called nursing. This typically begins when workers are about 3 to 10 days old, though the exact timing varies with colony needs and individual variation. Nurse bees feed larvae with nutritious secretions produced by glands in their heads. For the youngest larvae, this secretion is called royal jelly, a protein-rich substance that's fed to all larvae for the first few days. Later, most larvae receive a mixture of pollen, honey, and glandular secretions, while larvae destined to become queens continue receiving royal jelly throughout their development. The composition of larval food is precisely calibrated, and nurse bees make dozens or even hundreds of visits to each larva during its growth period, adjusting the amount and type of food as the larva develops. Nursing is demanding work. A colony with thousands of developing larvae requires constant attention from dozens or hundreds of nurse bees. Each larva grows rapidly, increasing its weight by more than 1,000-fold in just a few days, and all that growth comes from the food provided by nurses. The nurses themselves must consume large amounts of pollen to produce the glandular secretions they feed to larvae, creating a direct link between the colony's pollen stores and its ability to raise the next generation. Later, workers may become house bees, helping build comb and store food. This transition usually happens when workers are roughly 10 to 20 days old as their wax-producing glands reach peak activity. Wax building is a careful task that involves shaping small wax scales into the precise geometry of honeycomb. A builder bee takes a wax scale from her abdomen, chews it to make it pliable, and then adds it to the growing comb, using her mandibles and legs to press and shape it into place. Other bees may come along and further refine the work, smoothing walls and adjusting angles until the cell has the right dimensions. It's collaborative construction, with no blueprints or foremen, just many individuals working side by side and the structure emerging from their collective effort. House bees also receive nectar from returning foragers and process it into honey. This involves reducing the water content through evaporation and adding enzymes that transform the nectar's sugars. A house bee might take a load of nectar from a forager, hold it in her honey stomach for a while to add enzymes, and then deposit it into a cell. Later, she'll return to that cell and fan her wings to speed evaporation. She might move the nectar from cell to cell, spreading it out to increase surface area and accelerate drying. This process can take several days, and it requires constant attention from house bees who monitor the nectar's thickness and decide when it's ready to be capped with wax. Some workers guard the hive entrance, checking incoming bees to verify they belong to the colony. Guards position themselves near the entrance, antennae extended, and inspect every bee that tries to enter. They're looking for the colony's distinctive odour, a chemical signature composed of various pheromones, including the queen's, mixed with scents from the hive's wax and honey. Bees from the same colony share this odour, while bees from other colonies smell different. If a guard detects a foreign bee trying to enter, she'll block its path and, if necessary, grapple with it to drive it away. Guarding is most common among bees that are transitioning from house bee tasks to foraging, typically around 2 to 3 weeks old, though again, this varies with colony needs. Older workers often become foragers, leaving the hive to collect nectar, pollen, water, or resins. Foraging usually begins when workers are about 3 weeks old or older, and it continues for the rest of their lives, which might be only another week or two during busy times or several months if they emerge in late summer or fall. Foraging is the most dangerous work bees do. They face weather, predators, exhaustion, and the simple challenge of navigating to food sources that might be miles away and then finding their way home. Many foragers don't return from their flights, lost to birds, spiders, or simply wearing out their wings and falling to the ground far from the hive. This age-related shift in jobs helps the colony run smoothly without needing a strict schedule or central coordinator. Young bees, which have never left the hive and are still learning its layout and their nestmates' scents, do interior work where mistakes are less costly. Middle-aged bees, with fully developed glands and some experience, handle the complex tasks of nursing and building. Older bees, nearing the end of their lives anyway, take on the risky work of foraging. If a forager dies while out collecting food, the colony has lost a relatively old individual who might have died soon regardless. If the colony lost young nurses at the same rate, it would struggle to feed its larvae and maintain its population. This age-based division of labour isn't conscious strategy. Bees don't decide to protect the young. But it's an evolved pattern that improves colony survival, and that's good enough. With daily work flowing quietly through the hive, we can drift into the colony's communication systems, how bees share information in simple, gentle ways that allow thousands of individuals to coordinate without confusion. Honeybees communicate using smells, movement, and contact, creating a rich exchange of information that flows constantly through the hive. Chemical signals called pheromones can influence behaviour across the colony, even among bees that never directly encounter the source of the signal. These pheromones are produced by various glands and can signal different things depending on their composition and concentration. Some pheromones are short-lived, fading within minutes or hours, while others persist on the comb or on bees' bodies for days. The queen's pheromones help maintain cohesion and signal her presence throughout the colony. As mentioned earlier, the queen produces queen substance from her mandibular glands, a blend of several chemicals, the most important being 9-oxo-2-decenoic acid, or 9-ODA for short. Workers that come into contact with the queen pick up these pheromones through physical touch, then spread them to other bees through contact and through the food they share. In this way, the queen's chemical presence circulates through the entire hive, even reaching bees on combs that the queen hasn't visited in days. As long as this signal remains strong, workers can be confident the colony has a healthy, actively laying queen, and they continue with their normal tasks. If the signal weakens, it triggers a remarkable response. Workers begin building special queen cells and feeding the larvae inside with royal jelly, preparing to raise a new queen before the old one fails completely. Worker bees can release alarm pheromones to alert others to threats, though this system is carefully modulated to avoid panic. The most common alarm pheromone is released from a gland near the base of the sting and smells somewhat like bananas. Specifically, it contains isopentyl acetate, which is also a component of banana oil. When a bee stings a threat, she releases this pheromone, which attracts other bees and makes them more likely to sting as well. This can escalate a defensive response rapidly, but it's not a mindless reaction. Bees are more likely to respond to alarm pheromone when they're already on alert, for instance, near the hive entrance where guarding behaviour is expected, and less likely to respond when they're deep inside the hive engaged in nursing or building tasks. The system is sensitive but not hair-trigger, allowing the colony to mount an effective defence without constantly being on edge. Bees also share information by touching antennae and exchanging food, a behaviour called trophallaxis. When a forager returns with nectar, she'll often pass it to a house bee through mouth-to-mouth contact, and in that exchange, both chemical information and actual food are shared. The house bee gets a sample of the nectar's quality and the scents of the flowers the forager visited, information that might influence her own future foraging choices if she transitions to foraging herself. The forager, meanwhile, might receive a small amount of honey from the house bee, providing her with energy and also with a sample of what's currently stored in the hive. These exchanges create a network of information flow where bees sample each other's experiences and gradually develop a shared sense of the colony's state. The waggle dance is perhaps the most famous example of honeybee communication, a movement pattern bees use to indicate food sources to their nestmates. When a forager discovers a productive patch of flowers, she returns to the hive and, under the right conditions, performs this dance on the vertical comb inside the darkened hive. The dance consists of a figure-8 pattern, with the key information encoded in the middle portion, the waggle run. During this portion, the bee moves forward while waggling her body side to side, and it's the direction and duration of this waggle run that conveys information about the food source's location. The angle of the waggle run relative to straight up on the comb relates to the direction of the food relative to the sun. If the food is in the same direction as the sun, the bee waggles straight upward. If the food is 90 degrees to the right of the sun, she waggles 90 degrees to the right of vertical. The dance transposes the horizontal landscape outside into an angular code on the vertical comb, using gravity as a reference since the bees can't see the sun from inside the dark hive. It's an elegant solution to the problem of communicating three-dimensional spatial information in a completely different environment. The length of the waggle portion can relate to distance, though the relationship isn't perfectly linear. Generally, longer waggle runs indicate more distant food sources, with the bee spending more time waggling for flowers that are farther away. Recent research has shown that the exact dialect varies between honeybee subspecies and even between regions, with some bees waggling longer for the same distance than others. But within a colony, the system is consistent enough that bees can interpret each other's dances and successfully navigate to food sources they've never visited before, guided only by the information encoded in their nestmates' waggling movements. Other bees follow along with the dancing bee, touching her with their antennae and running behind her through the dance pattern. They're not just watching, they're gathering information through multiple senses, detecting vibrations, smells from the flowers clinging to the dancer's body, and possibly even sounds produced during the waggle run. After following several circuits of the dance, a follower may be recruited to visit the advertised food source. She'll fly out from the hive using the direction and distance information from the dance along with visual landmarks and the sun's position to navigate. If she successfully finds the flowers, she might return and perform her own dance, recruiting even more foragers in a positive feedback loop that focuses the colony's foraging effort on the most rewarding flower patches. Even without understanding every detail of the waggle dance or the complex chemistry of pheromones, it's soothing to know the hive has a calm language, a set of signals that allow thousands of individuals to share information and coordinate their activities without chaos. The hive's signals are like quiet notes passed between neighbours, keeping the day running smoothly without shouting, without confusion, without anyone needing to know everything. Each bee knows what she needs to know, shares what she can share, and the collective intelligence of the colony emerges from these small, repeated exchanges. Now, we'll step outside with the foragers, following the gentle path from flower to hive and back again, through the sun-warmed air and over landscapes painted with colour and scent. Forager bees visit flowers to collect nectar, a sweet liquid made by plants as an incentive for pollinators. Nectar is produced by special glands, called nectaries, usually located at the base of a flower where a visiting insect must brush past pollen-bearing stamens or pollen-receptive stigmas to reach it. The sugar concentration of nectar varies widely. Some flowers produce thin, watery nectar that's only 10 or 15% sugar, while others produce thick, syrupy nectar that might be 50 or 60% sugar. Bees show a preference for more concentrated nectar because it provides more energy for the same amount of effort, and they can sense sugar concentration using taste receptors in their mouth parts. A forager collects nectar by extending her long tongue, called a proboscis, and sucking the liquid into a special chamber in her body called the honey stomach or crop. This isn't her true digestive stomach. It's a storage tank that can be filled with nectar and later emptied back out. A bee can carry a load of nectar weighing nearly as much as her entire body, and when fully loaded she'll fly back to the hive heavy, and ungainly but driven by the promise of delivering valuable resources to her colony. Nectar is carried back to the hive and eventually becomes honey through a process of transformation that involves both chemical changes and physical evaporation. But before we dive into that process, let's stay with the forager a moment longer and consider the other resources she might collect. Foragers also collect pollen, which is a key protein source for the colony. While nectar provides carbohydrates, quick energy for flight and for maintaining body temperature, pollen provides the proteins, fats, vitamins and minerals needed for growth and development. Larvae consume large amounts of pollen, processed by nurse bees, and young adult bees also eat pollen to develop their glands and build up their strength. A colony without adequate pollen can't raise healthy brood, even if it has abundant honey, so pollen collection is absolutely essential. Pollen grains stick to bees as they move among flowers, clinging to the branched hairs that cover much of a bee's body. These hairs have a feathery structure at the microscopic level, providing lots of surface area for pollen to catch on. A bee visiting flowers quickly becomes dusted with pollen, and this accidental coating is what makes pollination possible. Some of that pollen rubs off on the next flower, potentially fertilizing it. But the bee also deliberately collects pollen by grooming herself in mid-flight, or while perched on a flower. She uses her legs to brush pollen from her head, body, and legs forward and downward, gradually accumulating it into the pollen baskets on her hind legs. Pollen baskets, or corbiculum, are smooth, slightly concave areas on the outer surface of a bee's hind legs. surrounded by long, curved hairs that help hold the pollen in place. The bee moistens the pollen with a bit of nectar or honey, making it sticky, then packs it into these baskets, building up two bright pellets that can be seen easily on a returning forager. The colour of these pollen pellets varies with the flowers being visited: bright yellow from dandelions, orange from poppies, blue-grey from phacelia, white from apple blossoms. A hive entrance during active foraging can look like a small airport, with bees arriving every few seconds sporting pollen loads of different colours, a visual testament to the diversity of flowers they're visiting. A forager may visit many flowers in one trip, sometimes dozens or even hundreds, depending on how far she's travelling and how rewarding each flower is. But here's an interesting behaviour. Bees often show flower constancy, focusing on one type of flower during a trip. A bee that starts her morning visiting clover will likely continue visiting clover for the rest of that foraging bout, even if she passes other flowers that might be equally rewarding. This behaviour has advantages for both the bee and the plants. For the bee, it means she becomes efficient at handling that particular flower type, learning exactly where the nectar is located and how to access it quickly. For the plants, it means the pollen being moved is more likely to reach a compatible flower of the same species, making pollination more successful. It's an unintentional partnership, but one that has shaped both flowering plants and their pollinators over millions of years. This flower constancy can make pollination more efficient for those plants, increasing the chance that pollen will reach the right destination rather than being wasted on incompatible species. From an evolutionary perspective, plants that could somehow encourage flower constancy in their pollinators would have an advantage, and we see various strategies that might promote this behaviour. Flowers might bloom in large patches, making it easy for a bee to move from one to the next without encountering other species. They might offer consistent rewards so bees learn they're reliable. They might have distinctive colours or scents that make them easy to recognise and remember. All of these factors might encourage a bee to keep visiting that flower type once she's started, and that benefits both parties. Bees use the sun and landmarks to help navigate across the landscape between hive and flowers. The sun is a reliable compass, moving predictably across the sky throughout the day, and bees have an internal clock that compensates for the sun's movement. A bee that flies north at 10 in the morning, when the sun is in the southeast, can fly north at 2 in the afternoon, when the sun is in the southwest, by adjusting the angle she flies relative to the sun. This time-compensated sun compass allows for accurate navigation even on foraging trips that last hours. Visual landmarks also play an important role, especially near the hive where precise navigation is crucial. Bees memorize the appearance of the landscape around their hive, trees, buildings, patterns of fields and forests, and use these landmarks to recognize when they're getting close to home. They seem to store multiple snapshots of the view from different distances, matching what they're currently seeing against these memorized images to gauge how far they've traveled along the familiar route. This ability has been demonstrated in elegant experiments where researchers change the landscape around a hive, moving a distinctively coloured panel or altering the arrangement of artificial landmarks, and show that bees adjust their flight paths based on these visual cues. A foraging trip can feel like a calm loop, out to the flowers, moving from bloom to bloom in the warm sunlight, filling her honey stomach with nectar or packing pollen into her baskets, then home to warmth and the familiar darkness of the hive, unloading her cargo to waiting house bees, perhaps taking a moment to feed herself from stored honey, and then out again if the day is still young and the flowers still producing nectar. On a good day, a forager might make 10 or more trips, covering dozens of miles total, visiting thousands of flowers and bringing back valuable resources that will feed the colony and be transformed into the long-lasting stores that will carry the hive through leaner times. With nectar and pollen returning home in a steady stream throughout the daylight hours, we can see how the hive turns these gifts into stored comfort: honey, pollen stores, and the waxen rooms that hold them. Honey begins as nectar but is changed through processing by worker bees, a transformation that converts a perishable liquid into a stable, long-lasting food source. The process starts as soon as nectar enters the hive. A forager doesn't deposit her nectar load directly into storage cells. Instead, she transfers it to a house bee through trophallaxis, that mouth-to-mouth food exchange we mentioned earlier. The house bee receives the nectar and begins the transformation process. Bees pass nectar between each other multiple times, and each time they hold it in their honey stomach for a while, mixing it with enzymes produced by glands in their head and thorax. The most important enzyme is invertase, which breaks down sucrose, the main sugar in most nectar, into glucose and fructose. This chemical change serves several purposes. First, glucose and fructose are sweeter than sucrose, making the final honey taste sweeter, even though the total sugar content might be similar. Second, glucose and fructose are simpler sugars that are easier for bees to digest and use for energy. Third, the breakdown of sucrose creates a more complex mixture of sugars that's less likely to crystallize quickly in storage. Some honeys do crystallize eventually. You've probably seen honey in a jar turn thick and grainy, but the enzyme treatment delays this and keeps the honey flowing longer. Another enzyme, glucose oxidase, produces hydrogen peroxide from glucose, giving honey mild antibacterial properties. This helps prevent fermentation and spoilage, allowing honey to be stored for months or even years without going bad. In fact, honey found in ancient Egyptian tombs thousands of years old was still chemically recognizable as honey, testament to its remarkable stability. While enzymes are being added, the nectar is also being concentrated. Fresh nectar might be 60 to 80% water, which is far too dilute to store safely. It would ferment as yeasts consume the sugars and produce alcohol and carbon dioxide. Honey, by contrast, is typically less than 20% water, often closer to 17 or 18%. Achieving this concentration requires removing most of the water, and bees accomplish this through evaporation. Bees fan their wings to help evaporate extra moisture from nectar in the comb, creating air currents that carry away water vapor. If you could see inside a hive during honey-making season, you'd find many bees stationed over cells of ripening nectar, their wings beating steadily, working in shifts to keep the air moving. The nectar might be spread across multiple cells initially to increase its surface area and speed evaporation. As it thickens, bees consolidate it, moving it from cell to cell and gradually filling fewer cells with denser liquid. When honey is thick enough, when it reaches that magic 18 to 20% water content, bees cap the cell with wax to seal it. The capping is a thin layer of wax, slightly lighter in colour than the comb itself, and it serves as a lid that keeps the honey from absorbing moisture from the air and also protects it from contamination. Capped honey can be stored almost indefinitely, providing a reserve of energy that will sustain the colony through winter when no flowers are blooming and no foraging is possible. Honey is a long-lasting energy source for the colony, but it's not the only food stored in the hive. Pollen is often stored in cells too, usually in frames near the brood nest where it's easily accessible to nurse bees who need it to produce larval food. Returning foragers scrape their pollen loads off their legs and pack them into cells, and house bees come along and pack it down more firmly, sometimes adding a bit of nectar or honey to help preserve it. This stored pollen is sometimes called bee bread, especially when it's been in the cells for a while and has undergone some fermentation. The fermentation process, driven by yeasts and bacteria naturally present on the pollen, actually makes the nutrients more available to bees, improving its nutritional value. Bee bread has a slightly tangy smell, different from fresh pollen, and it represents processed protein, ready to be consumed when needed. These stores, honey for carbohydrates, bee bread for protein, help feed larvae and support the colony during lean times. A strong colony going into winter might have 60 or 80 pounds of honey stored across its combs, enough to sustain the cluster through months of cold weather when flying is impossible. The bees will consume this honey slowly, converting it back to energy to maintain their body heat and to produce the small amounts of food needed for the winter-born larvae that will become the spring workforce. The hive's storage system is like a pantry built from careful repeating work, thousands of foraging trips, each bringing back a small amount of nectar or pollen, each load processed and stored with attention and care. There's no refrigeration, no modern preservation technology, just enzymes, evaporation and wax caps, yet the food stays good for months. The calm goal driving all this work is simple: enough food and warmth to keep the colony steady through whatever challenges the seasons bring. Now we'll drift into the next generation, how new bees grow in the comb's quiet hexagon rooms, from egg to larva to pupa to adult, in a cycle that renews the colony continuously. The queen lays eggs in individual cells of the honeycomb, moving methodically across the comb and pausing at each empty, cleaned cell to inspect it. She curves her abdomen down into the cell and deposits a single egg, attached to the bottom of the cell by a small adhesive surface. The egg is tiny, about the size of a grain of rice, pearly white and slightly curved. Each egg is placed carefully in the centre of the cell, standing upright at first, though it will tilt over in the following days. After a few days, typically 3, eggs hatch into larvae. The larva emerges from the egg shell and immediately begins its primary job, eating and growing. At this stage, the larva is a small, white, C-shaped grub with no legs, no wings, no eyes, just a simple body with a mouth at one end and a digestive system processing food as fast as it's provided. The larva is completely dependent on nurse bees for food, and they visit it frequently, depositing small amounts of food directly into the cell. Larvae are fed by worker bees, who visit them repeatedly, throughout the day, hundreds of times during the larva's development. For the first few days, all larvae receive royal jelly, that protein-rich secretion from the nurse bee's hypopharyngeal glands. Royal jelly is a creamy, slightly acidic substance containing proteins, sugars, fats, vitamins, and various trace compounds. It's incredibly nutritious, designed to support rapid growth. A larva eating royal jelly will increase its weight dramatically, growing from an invisible speck to a substantial grub in just a few days. The food for larvae can change as they grow, depending on what they're destined to become. Larvae that will develop into workers receive royal jelly for the first 2 to 3 days, and then their diet shifts to a mixture of royal jelly, honey, and pollen. This change in diet is what determines they'll become workers rather than queens. The proteins and other nutrients in this mixed diet support their development, but they don't receive the constant rich feeding that would allow them to develop into reproductive queens. Meanwhile, larvae in special queen cells, larger peanut-shaped cells that hang vertically from the comb, continue receiving pure royal jelly throughout their entire larval period. This continuous royal jelly diet triggers different developmental pathways, causing these larvae to develop functioning ovaries and other queen characteristics. It's remarkable that the same genetic material, eggs laid by the same queen, can develop into either workers or queens depending entirely on diet. This developmental flexibility, called polyphenism, allows the colony to produce the type of individuals it needs when it needs them. In normal times, with a healthy queen present, the colony raises workers. If the queen dies or weakens, workers can quickly raise a new queen from any recently laid egg by feeding the larva royal jelly instead of the standard worker diet. As larvae develop, they eventually fill their cells, their white bodies curled, into the hexagonal space, head toward the opening where nurse bees can easily feed them. After about 5 or 6 days of eating, the exact timing varies with the type of bee and environmental conditions, the larva stops feeding and prepares for its transformation. Worker bees cap the cell with wax, sealing the larva inside in a small, dark chamber. This capping is slightly more domed than the flat capping over honey cells, and experienced beekeepers can distinguish brood comb from honeycomb at a glance based on the pattern of cappings. Inside its sealed cell, the larva spins a thin silk cocoon, coating the cell walls with strands of silk produced from glands in its head. This cocoon provides a smooth surface and some protection during the vulnerable pupal stage to come. After completing the cocoon, the larva straightens out its body and enters the pupal stage. During the pupal stage, the bee's body forms into its adult shape through a process called metamorphosis. This transformation involves the breakdown and reorganization of larval tissues and the development of adult structures. Wings develop from small tissue buds, legs elongate and develop their adult segments, eyes form and darken, and internal systems are reconfigured for adult life. The pupa doesn't eat during this time. All the energy for this transformation comes from the food consumed during the larval stage. This is why adequate larval feeding is so critical. A poorly fed larva becomes a small, weak adult. The pupal stage lasts about 12 days for workers, a bit longer for drones and shorter for queens. During this time, the pupa remains motionless in its cell, visible only if the cell capping is removed. It starts out pale and soft, with its developing features barely distinguishable. Gradually it darkens, with the eyes turning from white to purple to black, the body taking on the tawny browns and blacks of an adult bee. When the bee is ready to emerge, it chews through the wax, capping from the inside, breaking free and climbing out onto the comb. The newly emerged adult is pale, fuzzy and slightly damp, with soft body parts that will harden over the next hours. Other bees mostly ignore the new arrival, who begins her adult life by wandering across the comb, familiarizing herself with her surroundings and soon starting on those early tasks of cleaning cells and helping with heating or cooling. The hive's nursery is quiet and steady, built on patient care and repetition. The queen lays eggs, hour after hour, day after day. Nurse bees make their rounds, feeding larvae on a schedule that requires precision and dedication. House bees cap cells when the larvae are ready, and later might help newly emerged bees clean off their wings and antennae. The process continues in overlapping waves, with cells at every stage of development scattered across the brood combs, creating a patchwork of eggs, young larvae, old larvae, capped pupae, and occasionally an empty cell, just cleaned and ready for the next egg. It's a production line without a conveyor belt, a nursery without nurses in the human sense, just many individuals doing what needs doing, and from that, new bees emerge daily to replenish the workforce. With the life cycle gently turning, bringing new bees into the world as older ones wear out and die, we can look at the hive in its wider world. Seasons, flowering times, and the long rhythm of the year that shapes everything the colony does. Honeybee activity often changes with the seasons, following the availability of flowers and the challenges of temperature. In temperate regions where winters are cold, this seasonal pattern is dramatic. In tropical or subtropical areas where flowers bloom year-round and temperatures stay moderate, the pattern is less pronounced, but it's still there, shaped by wet and dry seasons or by subtle shifts in which plants are flowering. In spring and summer, in a temperate climate, flowers are abundant and foraging increases dramatically. This is the season of growth. The queen ramps up her egg-laying in response to longer days and increasing food supplies, and the colony's population swells. A hive that emerged from winter with perhaps 10,000 bees might grow to 40 or 50,000 by midsummer. Every available worker is busy, foragers bringing in nectar and pollen, house bees processing and storing it, nurses feeding the growing larvae, builders adding new comb to accommodate both brood and stores. The hive entrance is a constant flow of arrivals and departures, and the overall sound of the hive rises to a deeper, fuller hum as the population peaks. During this time, the colony is building up its reserves for the future. Foragers are storing honey well beyond the colony's current needs, packing it into every available cell, and, if they're in a managed hive, filling the extra boxes that beekeepers add to provide more storage space. This surplus is what beekeepers harvest, taking the honey stored in these additional boxes while leaving enough in the main hive to sustain the bees through winter. A strong colony in a good location might produce 60 or 80 pounds of surplus honey in a season, or even more in exceptional years with abundant flowers and favourable weather. In colder months, bees may stay inside more and rely on stored honey. As fall approaches and flowers become scarcer, the colony begins to contract. The queen reduces her egg-laying, and the population starts to decline as older workers die off and aren't fully replaced. Drones are expelled, as we mentioned earlier, saving resources. Foraging opportunities become limited to a few late-blooming flowers, and eventually, as frost arrives, foraging stops almost entirely. The colony enters winter in a state of semi-dormancy. They don't truly hibernate. Individual bees remain active and alert, but their activity is restricted to the space inside the hive. They form a winter cluster, a tight ball of bees packed together for warmth, with the queen somewhere near the centre. The cluster can survive temperatures well below freezing outside, maintaining a core temperature of around 50 to 60 degrees Fahrenheit in the outer shell and warmer in the centre. Bees on the outside of the cluster face the cold, while those in the middle stay comfortable. As we discussed earlier, bees rotate positions to share the burden of being on the cold exterior. The cluster slowly consumes stored honey throughout the winter. Bees on the edge of the cluster, near the honey-filled cells, eat honey and share it through the cluster via trophallaxis. They generate heat through muscle activity. Those same flight muscles used for flying in summer now used for shivering, and that heat keeps the whole cluster warm enough to survive. On very cold days, the cluster contracts into a tighter ball to reduce heat loss. On milder days, it might expand, and bees might move to different areas of the comb to access fresh honey stores. The colony's population can grow during times of plentiful food, and this growth is strategic. More bees mean more foragers, which means more food can be collected, which supports even more bees. But this only works if the flowers are actually there to visit. A colony that expands too quickly in early spring, before many flowers are blooming, might exhaust its honey stores before new nectar is available, a dangerous situation called starvation in the midst of plenty, because while the landscape looks green and alive, there might not be enough nectar-producing flowers to feed a large population. When flowers are scarce, colonies may reduce activity to conserve energy. Fewer foragers go out because there's little to find. The queen reduces egg-laying because there's no point raising more bees if there isn't enough food to feed them. The colony enters a holding pattern, conserving resources and waiting for better conditions. This ability to scale activity, up and down based on resource availability, is crucial for long-term survival. Bees are sensitive to temperature, daylight, and food availability, and these factors interact in complex ways to regulate colony activity. Temperature affects flight. Bees generally don't fly when it's below about 50 degrees Fahrenheit, and even if it's warm enough, they won't fly in heavy rain or high winds, because these conditions make navigation difficult and increase the risk of being blown off course or grounded far from home. Daylight influences the queen's egg-laying and the colony's overall activity level, with longer days in spring triggering an increase in brood rearing. Food availability provides feedback. If foragers are returning with lots of nectar and pollen, the colony responds by raising more brood and expanding its workforce. If foragers are coming back empty, the colony contracts. Beekeepers sometimes help colonies with supportive care depending on region and season. They might provide supplemental feeding if the colony's honey stores are low going into winter, offering sugar syrup that bees can store in place of nectar. They might treat for pests and diseases, monitor the queen's health, and replace her if she's failing, add or remove boxes to give the colony appropriate space, or help manage swarming behaviour. Good beekeeping works with the colony's natural patterns rather than against them, supporting the bee's own instincts and abilities. The hive is a gentle example of adaptation, responding to change without panic, ramping up activity when conditions are good, and scaling back when they're not, all without stress or emergency meetings or contingency plans. Like the seasons themselves, the hive's rhythm is a return. Bloom, gather, store, rest, and then bloom again when warmth returns. There's something deeply reassuring about that predictable cycle. Knowing that the same patterns that sustained honeybee colonies for millions of years continue today in hives both wild and managed across the changing landscape. To finish this gentle journey through the honeybee's world, we'll bring the hive's steady routine back to a bedtime scale. Small, soothing lessons about calm repetition and shared warmth that we can carry with us into sleep. A honeybee colony works through many small actions repeated patiently. No bee decides the grand strategy or envisions the hive's future. Each bee responds to what's immediately around her. The smell of alarm pheromone that suggests a threat, the taste of incoming nectar that needs processing, the feel of an empty cell that could use cleaning, the hunger of a larva that needs feeding. These small local responses made by thousands of individuals create colony-level patterns that are remarkably sophisticated. The hive builds comb without an architect, stores food without a logistics manager, and raises young without a nursery administrator. It works because each bee is attentive to her immediate environment and willing to contribute what she can. No single bee does everything. The calm strength comes from teamwork. A forager who spends her days visiting flowers might never feed a larva or build a cell of comb. A nurse bee who tends larvae might never see the outside of the hive until she transitions to foraging late in life. A drone has no sting, no pollen baskets, no wax glands. He's specialised for mating and contributes nothing to the daily maintenance tasks. And yet the colony succeeds because all these different roles mesh together. The forager's nectar sustains the nurse who feeds the larva that becomes the next generation of workers. The drone, though he does no work in the hive, carries the colony's genes to other colonies, contributing to the broader genetic health of honeybee populations. Everyone has a role, and no role is wasted. The hive stays warm because many bodies cooperate in simple ways. One bee cannot heat the hive on her own, but 10,000 bees, clustering together, create a warm, stable environment that can persist through months of freezing weather. Each bee just needs to maintain her own body temperature and stay close to her neighbours. From that simple behaviour, collective warmth emerges. It's a reminder that sometimes the most impressive outcomes come from simple rules, followed by many individuals rather than from complex planning followed by a few. The honeycomb's repeating pattern reflects the colony's steady rhythm. Hexagon after hexagon, cell after cell, The comb extends through the hive in sheets of golden wax. Each cell is roughly the same size, oriented at the same slight upward angle, part of the same continuous structure. This repetition isn't boring, it's calming. It's evidence of work done patiently, of a task taken up by many workers across many days, each adding her small contribution to the growing whole. Looking at a frame of honeycomb, you're seeing the physical manifestation of cooperation, the accumulation of countless small efforts into something larger and more enduring. Foraging is a gentle cycle of leaving and returning, like a quiet breath. Out into the bright world of flowers and sun, into spaces vast and varied, then back to the dark, enclosed safety of the hive. Out again and back again. Each trip brings a small amount of nectar or pollen, never enough to feed the colony on its own, but enough to matter when multiplied by thousands of trips across hundreds of foragers throughout the day. It's the rhythm of gathering and returning home, a pattern as old as life itself, played out in miniature by creatures who navigate by the sun and remember the scent of flowers. The waggle dance shows that communication can be soft and efficient. There's no shouting, no desperate scrambling to share information, just a bee moving in a particular pattern on the comb, her movements encoding direction and distance, while other bees follow along and learn. The information flows through, the hive in waves, not all at once, not to everyone, but to those who need it and are ready to receive it. It's communication without urgency, sharing without pressure, and it works beautifully. Honey is a slow product of time, flowers, and patient work. It cannot be rushed. The nectar must be collected flower by flower. It must be passed between bees treated with enzymes, spread across cells, and fanned until the water evaporates away. Only then, when it's thick and concentrated and stable, can it be sealed in wax and stored. Each jar of honey represents thousands of flower visits, hundreds of hours of wing fanning, and days or weeks of processing. Its sweetness earned through patience, and somehow that makes it taste even better. The hive reminds us that organised life can feel calm, not rushed. We often think of organisation as requiring stress, deadlines, schedules, management, pressure. But the hive achieves extraordinary organisation without any of that. There's no schedule, just readiness to respond. There's no management, just individual bees making good decisions based on local information. There's no pressure, just the steady accumulation of small efforts over time. The hive is organised because its members are attentive, cooperative, and willing to work. That's enough. Tonight you can borrow that steadiness, one small breath, one small moment, repeated. You don't need to solve everything at once or plan out every detail of tomorrow. You just need to be here, now, settling into this moment and then the next one. Like a bee adding one cell to the comb, or making one foraging trip or feeding one larva, you can take things one step at a time, trusting that small efforts accumulate into something meaningful. And like a hive settling at night, your thoughts can quiet down and rest in warmth. The bees cluster together in the darkness, generating their own heat, creating their own stability, without needing the sun or the flowers or any external source of comfort. They have each other, they have their stores of honey, and they have the patterns of behaviour that have sustained their kind for countless generations. That's enough to carry them through the night and into the next day. You too have everything you need for this night. Your body knows how to rest, your breath knows how to slow, your mind knows how to let go of the day's concerns and drift into the gentle emptiness of sleep. You don't have to do anything except allow it to happen. Let your shoulders drop, let your jaw relax, let your breathing deepen and slow. Take a slow inhale, feeling your lungs expand, feeling air flowing in, bringing freshness and calm. And then a longer exhale, feeling tension flow out, feeling yourself sinking deeper into stillness. And imagine your mind becoming a quiet hive, warm, steady, and softly at peace. The day's tasks are done. The foragers have returned, the stores are sealed, the young are fed, the cluster has formed, everything is in its place, and night has settled over the hive with gentle certainty. There's nothing left to do tonight except rest, held in the warmth of this moment, supported by the simple knowledge that everything continues, that the patterns endure, that tomorrow will come when it comes and bring with it new flowers, new work, new, small moments of contribution and connection. But for now, there is only this: the darkness, the warmth, the quiet hum of existence continuing at its own pace, needing nothing from you except your presence, your rest, your slow drift into sleep. Breathe in the calm, breathe out the day, let yourself settle like bees in their winter cluster, finding your own warmth, your own steadiness, your own gentle rhythm that will carry you through this night and into the morning. The hive knows how to rest, and so do you. The hive knows how to endure, and so do you. The hive knows that tomorrow will take care of itself if tonight is spent in restful peace, and so you can let go now, releasing your hold on wakefulness, allowing sleep to arrive in its own time like a forager returning home, inevitable and welcome. Your breathing continues, steady and slow. Your heartbeat continues, reliable and calm. And your awareness gradually fades, becoming softer, quieter, more distant, like the sound of bees humming in a hive heard from far away, a gentle background to the great settling into rest that happens now, easily, naturally, like night falling over fields of flowers, like a hive wrapping itself in darkness, like the quiet satisfaction of work well done and warmth well shared. Rest now. The hive rests, the flowers rest, the day rests, and you can rest too, held in this moment, safe in this darkness, at peace in this quietness that deepens and deepens until there is nothing left but sleep. The next story is about to begin. Please relax and enjoy.